Chaitanya. Today we will be discussing the AITS test number 2. So let's begin the session. The first question is, golden rice is a transgenic crop which is insect resistant? No. Herbicide resistant? No. Rich in hirudin? No. Rich in beta carotene? Yes. So option 4 is the correct option. Moving ahead. Choose the mismatch from the following. F2 genotypic ratio of monohybrid cross with respect to complete dominance. So when we are talking about complete dominance, then what would be the genotypic ratio in garden P, right? So you know that the F2 genotypic ratio is 1:2:1 in case of a monohybrid cross which is showing complete dominance. Next is, so this is the correct option, it is not a mismatch. Next, F2 genotypic ratio of a monohybrid cross with respect to incomplete dominance in dog flower, that is Anthrynum majus, is 1:2:1. Again, this is a correct statement. In case of incomplete dominance, bacha, you have to keep in mind that the genotypic ratio will be equal to the phenotypic ratio, which will be equal to 1:2:1. Next, F2 genotypic ratio of monohybrid cross with respect to codominance in lentil is 1:2:1. Again, this is the correct statement, right? Again, in codominance, you can observe 1:2:1 as the genotypic ratio. Next, F2 genotypic ratio in dihybrid cross in garden P is 9:3:3:1. No beta, if we are talking about the F2 genotypic ratio, what is given here, it is not the genotypic ratio bacha. What is given is actually the phenotypic ratio, 9:3:3:1. But what is the genotypic ratio? 1:2:1:2:4:2:1:2:1. So this is the mismatch, right? So option 4 is the correct option as it is a mismatch. Moving ahead. Stirred tank bioreactors have been designed for. What are they designed for? They are designed for better availability of oxygen throughout the bioreactor. Yes, I have a beautiful diagram for this. So let's first mark the correct option. It is option number 4. Look at this diagram. So this is a stirred tank bioreactor. This bioreactor is designed in such a way that there is ample amount of oxygen availability, right? Moving ahead. Identify the correct sequence of events in rDNA technology. So whenever you are performing rDNA technology, students, you have to keep in mind that the first step would be what? Isolation of pure DNA, correct? Next, what will you do? You will ligate that desired DNA fragment within a suitable host, right? So then it will be 3. Next, what will you do? Insertion of rDNA into the host. Again, this is the next step. And once that is done, what would you do? You would go for extraction of the desired gene. So this is 1. So the sequence would be 2, 3, 4, 1, right? So is there anything like that? Yes. Second option, 2, 3, 4, 1 is the correct answer. So this is the vector where you are putting the desired gene of interest and then you are getting the recombinant DNA, right? This is how rDNA technology actually is working. Next, statement 1, Bt cotton is mainly resistant to pest. Yes, this is correct. Statement 2, Bt cotton is a GM plant, that is a genetically modified plant. Absolutely, this is also correct. So both A or statement 1 and statement 2 will be correct, right? So both the statements are correct. Next, so see, this is what happens. The Bt gene, it is incorporated within the host, right, within the DNA, right, and then what will happen? The cotton plant, there can be Bt cotton, Bt brinjal, so on, so they will become resistant to the insect pest. Now, next question, from the given list of crops, how many are semi-dwarf varieties of rice developed in India? Semi-dwarf varieties of rice, which are they? It is Jaya and Ratna. What is Kalyan Sona and Sonalika? They are varieties of wheat. What is IR8? IR8 is developed in Philippines, not developed in India, though it is a rice variety, but here the question clearly states developed in India. So your answer would be only 2, that is, this is the correct option. Is it clear? Jaya and Ratna, they are the semi-dwarf variety of rice developed in India. That is important here. You cannot consider IR8 here because it is not developed in India. Next we have match the following. Let me tell you, in NEET you get a lot of match the following types, and it's relatively easy to answer match the following also. So first is cowpea. Cowpea is Pusa Komal, correct. Next is rapeseed mustard, that is Brassica, it is Pusa Gaurav. Next, bhindi, Prabhani Kranti, right. So it is this. Wheat is Himgiri variety. So 4, 3, 2, 1, 4, 3, 2, 1. First option is the correct one. Next, an example of leaf modification is Phylloclad of Opuntia. No, Phylloclad is a stem modification. Leaf modification is phyllodes. So the next option, which is phyllodes of Acacia, is the correct one. So what exactly is a phyllodes? So in case of Australian acacia, what happens is that the leaflets are short-lived, and it is the petiole of the leaf that will become swollen and it will become photosynthetic. Such a swollen photosynthetic petiole in Australian acacia would be known as phyllodes. Clear? Moving ahead, select mismatch from the following. First is methanobacteria. It is used for biogas production. Absolutely correct. Next is Aspergillus niger. It is used for citric acid production. This is also correct. Clostridium butylicum, butyric acid production. Absolutely correct. Staphylococcus penicillin antibiotic production. No, penicillin is produced from Penicillium notatum or Penicillium chrysogenum, which is a fungus. So this is the mismatch, bacha, right? Next, the technology of biogas production was developed in India mainly due to the efforts of who were the two organizations involved here, IARI and Khadi and Village Industry Commission. So IARI and KVIC, these are the two institutes who were involved in the development of the technology of biogas production. Next, select the correct statement from the following. Antibiotics are anti-life to human beings. No, absolutely not. Had they been anti-life to human beings, we wouldn't have used it, correct? Next, probiotics are pro-life to harmful microorganisms. Absolutely not. They are not pro-life to the harmful microorganisms. Roquefort cheese is mainly ripened by growing a specific bacteria on them which give them a particular flavor. Now, Roquefort cheese is developed by Penicillium roqueforti. So it's a fungus and not a bacteria. So this is again an incorrect statement. Next, the dough which is used for making dosa and idli is fermented by bacteria. Yes, students. So this is the correct statement, right? Next, formation of one molecule of NH3 by nitrogenase requires input of. So if you remember, to fix one molecule of nitrogen, there is the requirement of 16 ATP, and ultimately this one molecule of nitrogen gets converted into 2 molecules of ammonia. But now the question says how many ATP would be required for the production of not 2 but only 1 molecule of NH3. So for the production of 2 molecules of NH3, 16 ATP is required. Therefore, for the production of 1 molecule of NH3, there would be the requirement of only 8 ATP. So second option is the correct option. Moving ahead, which plant is this? First you have to tell me before we go into the question. I think all of you have guessed it right. It is sphagnum, right? So identify A and B from the given figure. What is A? This is Antheridium. What is B? This is Archegonium. So A is the antheridial, like not Archegonium or Antheridium, it's the antheridial branch, and this is the archegonial branch, right? So antheridial branch and archegonial branch of sphagnum. This is the correct option. Now, let us talk again. We have a match the following. So many match the followings are there, so that your NEET examination becomes very easy. Okay. So pteris, what is it? It is having a monoecious prothallus. Okay. Pinus, you already know, needle-like leaves. Eucalyptus, it is an angiosperm, so it will have double fertilization. Selaginella, it is a heterosporous pteridophyte, and you know that heterospory is the precursor of seed habit. So here it will be precursor of seed habit. So 2, 1, 3, 4. Do we have something like that? 2, 1, 3, 4. So option 3 is the correct option. Next. Identify the correct sequence of taxonomic categories in Linnaeus hierarchy. I think this is the easiest question that you have in your what should I say, in this paper, right? I think all of us know kingdom, phylum or division, class, order, family, genus, species, right? So how does it go? See which one matches. So your phylum, they did not mention about, okay, so then after phylum is class, that is correct, then there is order, correct, then there is family, correct, genus and species. So option 4 is the correct option. They did not mention here about kingdom, right? So phylum, class, order, family, genus and species. Next, again another match the following. Fungus, so here there is a disease, fungus, it causes wheat rust, right, rust and smut. Caused by fungus, rust caused by Puccinia, smut caused by Ustilago, right? Next is viroid. Viroid causes potato spindle tuber disease. Then you have a bacterium which causes citrus canker. If you remember, right, Xanthomonas citri causes citrus canker. Then protozoan, sleeping sickness. Trypanosoma, it causes sleeping sickness, right? So it is 2, 3, 5, 1. 2, 3, 5, 1. Correct? Second option is the correct option. Moving ahead, which of the following is not a photosynthetic protist? Diatom, it is a photosynthetic protist. Dinoflagellate is also a photosynthetic protist, but blue-green algae, bacha, it is a cyanobacteria. It is a prokaryote, not an unicellular eukaryotic organism. So this is the correct option. This is not a photosynthetic protist. Moving ahead, in which of the following plant flowers can be divided into two similar halves by any vertical plane passing through the center, right, which cannot be, I'm so sorry, it is cannot be divided, correct? So that means we are talking about asymmetric flower. Which flower amongst these are asymmetric? I think all of you have guessed it right, it is canna, right? So this is a very clear line from the NCERT taken. Now in the Lac Operon I gene codes for, you all know this, come on guys, it is repressor protein, correct? So this is the correct answer. Moving on, so see, this is a Lac Operon that you have, so there is the RNA polymerase. This RNA polymerase, it will be transcribing Lac I and translating it to form the active repressor. This active repressor, in the absence of lactose, what would happen? This active repressor would be binding to the operator and not allowing RNA polymerase to transcribe the Lac, Z, Y and A genes, right? So now let's talk about the next one. Next question, which is polyembryony is found in? It is found in case of oranges, it is found in case of cycas as well. So both 1 and 2 is the correct option. Next, phloem element found in dicotyledonous plants are, in dicots, what are the phloem element that you find? You find sieve tube cells, you find companion cells, you find phloem fiber and you find phloem parenchyma. So which one would be the correct option from here? So it is sieve tube cells and companion cells, correct? Chalo. So phloem is the conducting tissue. You know that phloem helps in bidirectional transport and these are the elements of the phloem in a dicot, which are sieve tube elements, companion cells, phloem parenchyma and phloem fiber. Now, the genetic material of TMV is, what is it? Single-stranded RNA. All of you, I hope you remember this, right? Direct question. So here, as you can see, that viral host, if a virus is affecting plants, mostly it is single-stranded RNA and we are talking here about the tobacco mosaic viruses. Next, again another match the following, dicot root. Dicot root, whenever we are talking about root, so they have exarch xylem, correct? Exact xylem, it is present in the root. Endart xylem, it is present in the stem. Next, dicot stem, they have open vascular bundles. Absolutely correct. So here I'll write 5. Monocot leaf, they have mentioned grass in brackets. So grass, they have specialized cells which are known as the bulliform cells, also known as the motor cells, which helps in the rolling of the grass leaves, right? Next is monocot stem, right? Monocot stem, what will they have? They will have water containing cavities. So the answer would be 2, 5, 1, 4. Do we have something like that? 2, 5. So this one, 2, 5, 1, 4. Option 4 is the correct option. Next, in cyanobacteria, photosynthetic pigments are found in. Come on, again a direct statement from the NCERT, which is the chromatophores, correct? So chromatophores are certain structures which would be filled with photosynthetic pigments in bacteria like the cyanobacteria, right? Which cell organelle divides the intracellular space of the cell into 2 distinct compartments? Yes, I think if you have read endoplasmic reticulum properly, you would be able to answer this question. The endoplasmic reticulum divides the cell into 2 compartments. The part of the part which is within the endoplasmic reticulum, it is known as the luminal space, and the part which is outside the endoplasmic reticulum, it is known as the extraluminal space, right? So it is the endoplasmic reticulum that divides the cell into 2 compartments, the luminal space within the ER, in the space which is outside the ER, that is the extraluminal space. Next, application of which phytohormone removes inhibitory substance from the seed? I think you again know this answer. The answer is gibberellin, correct? Chalo. Now, identify the correct combination. AUG, yes, it codes for methionine. UAG, it is a stop codon. UGG, tyrosine. Do you think it's correct? No, it is not correct. UGG does not code for tyrosine, rather it codes for tryptophan, and GUG, you know that it codes for valine. So this option 3 is incorrect match. UGG stands for or codes for tryptophan, right? So please see here clearly, UGG, it codes for tryptophan and not tyrosine. Now, again another match the following here. So, synapses. When does synapses formation take place? In prophase 1. I will straight away write 4. Splitting of the centromere. During meiosis, when does the centromere split? During anaphase 2, obviously, right? Next, nuclear membrane and nucleolus reappear. When will it reappear? In telophase 2. So, I will write 5 here. Next, microtubules from the opposite poles attached to the kinetochores of the homologous chromosome. When does this happen? In metaphase 1, right? This one. So, 4, 2, 5, 1. 4, 2, 5, 1. This is the correct option. So here we have a little bit of explanation that the bivalent align at the equatorial plate, microtubules of the spindle attached to the pair of the homologous chromosome. So if you can clearly see here, right? Clearly see what happens here, right? Next, anaphase 2. In anaphase 2, what will happen? See, splitting of centromere will take place during anaphase 2. In anaphase 1, what happens? In anaphase 1, the spindle fibers, they will polymerize, depolymerize, they will shorten in length, and therefore the homologous chromosomes will be separated. Correct? Now, next one, let us talk about the next question, which is primary CO2 acceptor in C3 plant is. What is it? In C3 plant, it is a pentose ketose sugar. Again, you have to read the lines of NCERT. It is there that IUBP, ribulose 1,5-bisphosphate. It is a pentose sugar, 5-carbon sugar, and it is a ketose sugar. It's clearly mentioned in the NCERT. So, it is pentose ketose sugar, and what is the name of the sugar? It is RUBP, ribulose 1,5-bisphosphate. Next, itario of alkenes are found in. This is beyond NCERT, but yes, you can get 1 or 2 questions beyond NCERT. This is from morphology. So, the answer would be strawberry. Correct? Next, wind pollination is common in. It is common in grasses. It is also common in gymnosperms. So, both 1 and 2 would be the correct option. Next, monocarpic perennial plant is, monocarpic perennial plant, it is what? Bamboo, first option. Can you name another monocarpic perennial plant? I think if you have read your syllabus properly, you know that the answer is Neelakuranji or Strobilanthus kuntiana, which is monocarpic perennial, and it flowers once in every 12 years. Last time it flowered was in the month of September, October 2018, right? Moving ahead. So now let us talk about codominance. Okay? So codominance, so again another match the following actually. Codominance, it is found in the AB blood group in the humans, correct? Yes, AB blood group, IA, IB, both of them will be expressed, and therefore we have the AB blood group, and that is what codominance tell us, right? So both the alleles will be capable of expressing themselves, right? So codominance 4. Incomplete dominance, where do you find it in? In the flower color, in dog flower, in heterozygous condition. So yes, let us say that capital R if it is red, small r if it is white. Then in heterozygous condition, that is capital R, small r, it will be bacha what? It will be pink, right? So this is representing what? It is representing incomplete dominance, right? So this is 2. Polygenic inheritance, all of us have read it so many times that it is skin color of man. And then there is linkage. Yes, linkage, eye color and body color genes on X chromosome in Drosophila, they are linked genes and they will show linkage, right? So it is 4-2-1-3, 4-2-1-3. Third option is the correct answer. Next, which of the following is immobile and divalent cation within plants? Which one is it? Calcium, right? I think all of you know this, it is calcium. Direct question again from the NCERT. Xylem and phloem transport in plants respectively are. This paper is very easy, right, for you all? I think all of you must have answered this question. What is it? Xylem, it is unidirectional, phloem it is bidirectional. First one is the correct answer. Very good. Let's move ahead. Again another match the following. You might be wondering why so many match the following. Then I would advise you please go back and see the last year's NEET paper. You will be totally convinced as to why there are so many match the followings here. Right. So chlorine, it is involved in photolysis of water. Please remember that along with manganese, chlorine is also involved in the photolysis of water. So I'll write 4 here. Boron, you all know that it is involved in the germination of the pollen tube, right? Next is magnesium. Magnesium, it is occupying up in the porphyrin ring structure of the chlorophyll. Yes, it is present in the porphyrin ring structure of the chlorophyll. And then you know that there is the sulfur containing amino acid which is methionine, right? Methionine contains sulfur. So what would be the answer? 4-1-2-3. 4-1-2-3. First option. Badhiya. Next. Volvox is a colonial motile flagellated green algae. Very direct question. Now, how many chromosomes are found in the root tip cell during metaphase stage? So during metaphase stage also the number of chromosomes in the root tip would be remaining as 2n, correct? And 2n in case of onion is 16. So option 4 is the correct answer. Next, in the citric acid cycle, GTP molecule is synthesized during the conversion of. When is GTP synthesized in the Krebs cycle, bacha? When the succinyl coenzyme A is converted to succinic acid, that is the time where GTP would be synthesized, correct? Next, which of the following is not a physiological effect of auxin? It helps or it induces xylem differentiation. Absolutely correct, it does help. It induces parthenocarpy, absolutely. If you spray the stigma of a flower with auxin, it gets the same stimulus as if pollination has taken place, and the ovary will develop into the fruit, but the ovules will not develop into the seeds, and therefore you will get parthenocarpic fruits, right? So this is absolutely correct. It removes apical dominance. No, it does not remove, it rather promotes apical dominance. So this is the incorrect statement. That is what they are asking, which is not a physiological effect of auxin. I want all my students to note these: not, do not, incorrect, correct. Don't get confused here. right? Next, statement 1, lipids are strictly macromolecules. Is it correct? No, they are not macromolecules. Though they are found in the acid insoluble fraction, still they are what? They are micromolecules. Next, acid soluble fraction have polysaccharides, proteins, nucleic acids and lipids. No, just now I told it is acid insoluble. So in this case, both statement 1 and statement 2 are incorrect. So this is the correct option. Next, identify the mismatch from the following. Periderm, phloem, phelogen, pheloderm, absolutely correct. Sapwood, secondary xylem mainly, which helps in the conduction of water and minerals. Yes, correct. Early wood, it is sapwood, which is lighter in color. Fine. Bark, all the tissues interior to the vascular cambium. No, no, no. It is exterior to the vascular cambium. Correct. So this is the bark. Okay. So this is the incorrect or the mismatched. Which type of nucleic acid is known as the adapter molecule? What would be the answer? You all know it is the tRNA or the transfer RNA. Now, tetramerous, polysyphilis, polypetalous and tetradynamous flowers are seen in. These are specific for the Brassicaceae family, so it is seen in case of Brassica. We have a slide on this. So see, what do you mean by tetradynamous? 4 plus 2 arrangement of stamens. So see, 1, 2, 3, 4. 4 inner stamens would be long, 2 outer stamens would be short. This arrangement of stamens is known as a tetradynamous condition, characteristics of the Brassicaceae family, right? Next, embryo sac is formed from a, where is it formed from? From a megaspore, simple, right? Next, how many of the following are ornamental plants? Mulethi? No, it is a medicinal plant. Lupin? Yes. Gloriosa? Yes. Petunia? Yes. Sweet potato? Yes. And tulip? Yes. So how many? Yes. 1, 2, 3, 4, 5. So 5 only. Correct? Moving ahead. Now, which of the following statement is wrong with respect to fruit flies? Which is wrong? They complete their life cycle in about 2 weeks. Is this wrong? No, this is correct. They could be grown in a simple synthetic medium in the laboratory. Yes, for that. This was one of the main reason why Morgan had chosen Drosophila, because it's very easy to grow them, right? Male flies are bigger than female flies. No, no, no. Male flies are smaller than the female flies. So this is the incorrect or the wrong statement. So this is the fruit fly. Now, which of the following restriction enzymes produce blunt ends? It is which one? HIND2 and EcoR5. Yes, so both 1 and 2. EcoR1, it produces sticky ends and not blunt ends. Next, identify A and B from the given figure. What is A? It is the collar of the bacteriophage. What is B? It is the tail fiber or the prongs of the bacteriophage. So it is option 3, collar and prongs. Next, phytoplanktons are pioneer in this succession. Phytoplankton, where are they pioneer in? They are pioneer in hydrocerae, right? For xerocerae, it is the lichens which is the pioneer. So, phytoplanktons, they are the pioneers for xerocerae, right? So, students, that is all for this session. I will again meet you in the next session. Thank you so much.
Hello dear students, this is Dr. Priyanka and now it's time for zoology. So now we will discuss the questions of zoology. Let's see what is the first question on your screen. So first question on your screen is, which evolutionary force is related to the founder's effect in a population? Now here you can see that founder's effect in a population is related to genetic drift. Okay, and what is genetic drift? Let's understand. Genetic drift is the sudden change in the gene frequency. So the sudden change in the gene frequency is known as genetic drift. Okay, it could be due to founder's effect if some individuals of a particular population moves to some other area and reproduce there, making another population B, the gene pool of the new population will be different from the original population. That is what is founder's effect. And the individuals that have moved to a different location, those are the founders for this population B. The another is bottleneck effect. If due to any natural calamity, many individuals of a particular population die, then what will happen? The other individuals will form a population, and again the new population will be different from the original one, and this is the bottleneck effect. So genetic drift can be of two types. One is the founder's effect that was asked in the question. We have seen this. And the another one is bottleneck effect, right? So two types of genetic drift are there. One is the bottleneck effect, another one is founder's effect. Now in the options we have natural selection, frame shift mutation and competition as well. So these are not the evolutionary forces that are related to the founder's effect. Now let's move on to our next question. Next question on your screen. Which of the following is correct match with reference to the population interaction? So here we have commensalism, balanus and whale, predation, ticks and dogs, mutualism, mycorrhiza and tree, amencialism, cuscuta and hedge plant. So we need to pick the correct combination. So the correct combination is regarding mutualism. So dear students, in mutualism there is positive interaction between two species A and B, right? That means both the species are benefited. Okay, example is mycorrhiza. Now mycorrhiza is a fungi that is associated with the roots of the higher plants, right? So mycorrhiza is actually a symbiotic relationship between the fungi and the roots of higher plants. Both are benefited in this relationship, right? Now fungi receives food made by the tree, and what fungi do? It increases the surface area for absorption of nutrients and water by the tree, right? So it is mutualism. So correct option here is 3. Here you can see population interaction in mutualism, there is plus sign, that means both species A and B. Both species A and B are benefited. In predation, one species is benefited, yeah, right? If a lion eats a deer, then deer is the one who is affected adversely, but the lion is benefited. So this is predation. Now let's move on to our next question. What does GPP in the equation GPP minus R is equal to NPP represent? Let's see all the options. The available energy for the heterotrophs, organic matter produced during photosynthesis, respiratory loss and primary productivity, or the energy transferred to the herbivores. See, they are asking about GPP. GPP stands for gross primary productivity. Okay, so GPP stands for gross primary productivity. And what is this GPP? This GPP is the organic matter produced during photosynthesis, right? So whatever organic matter is produced at the time of photosynthesis is GPP. Some of that organic matter will be used in respiration for deriving energy by the organism, right? So whatever is left is actually NPP. NPP is net primary productivity. Okay. Now let's move on to question number 54. The receptors for antibodies in myasthenia gravis occurs on? First, let's understand what is myasthenia gravis. Myasthenia gravis is an autoimmune disorder, right? So, myasthenia gravis is an autoimmune disorder. Okay. So, in myasthenia gravis, there is autoimmunity. That means our body's own immune system start attacking our own cells. So, there is autoimmunity against the receptors. The receptors, very important. And where these receptors are present? On the neuromuscular junction of, in the sarcolemma of skeletal muscle fiber. In the sarcolemma of skeletal muscle fiber. Okay. Understand that if on the sarcolemma there are receptors present for the neurotransmitter acetylcholine, we will study in detail. Okay. So, the receptors are present on the sarcolemma of skeletal muscle fiber. Okay. Understand, if these acetylcholine receptors which are present on the skeletal muscles are destroyed by our body's own immune system, no acetylcholine will be able to bind to it. As a result, there will be no generation of action potential in the muscles, skeletal muscles. Okay. So, ultimately, the impulses will not be able to cause contraction in the muscle fiber, and slowly there will be degeneration of the muscles. Okay. Let's understand this in detail. Okay. Here you can see, if this is the sarcolemma of the skeletal muscle fiber, there are receptors, okay, to which acetylcholine should bind, okay, which will come from the nerve endings, right? But if our body's immune system or the antibodies will destroy these receptors, now acetylcholine will not be able to bind to these receptors, and it will lead to degeneration of the skeletal muscles gradually. Fine. Now let's move on to our next question. Which of the following are seen in the Graphene Follicle of humans? Primary oocyte, first polar body; secondary oocyte, second polar body; ovum, second polar body; or secondary oocyte, first polar body. So please recall Graphene Follicle. Graphene Follicle is the mature. Graphene Follicle was the mature follicle that ruptures to cause ovulation. Okay, so it ruptures to cause ovulation. And which structure is released at the time of ovulation? It is the secondary oocyte. And during the formation of secondary oocyte, there is formation of one polar body as well. Okay. So, what will be the correct option here? The correct option will be secondary oocyte and the first polar body. So, at the time of ovulation, there will be release of secondary oocyte, and since meiosis 1 has completed, that means there will be formation of the first polar body. Okay. Let's move on to our next question. Before this, you can see this is the image of ovary. Here you can say this is primary oocyte. Ultimately, there will be formation of secondary oocyte, then tertiary oocyte, and finally there will be formation of Graphene follicle. Graphene follicle will have secondary oocyte that will be released at the time of ovulation. So if here it is Graphene follicle, it will release secondary oocyte at the time of ovulation. Okay. Now let's move on to our next question, which is question number 56. Match the following type of questions. Let's match it. Filariasis, in list 1 you have various diseases, and in list 2 you have causative organisms. Filariasis, also known as wucheriasis, is caused by infection of Wuchereria bancrofti and Wuchereria malai. These are the roundworms. These are the roundworms or ascalmins or nematoda. They have affinity for your lymphatic ducts, so because of which they accumulate in the lymphatics. There is accumulation of lymph in the lower limb and even in the genital area. So the lower limb is deformed like the foot of an elephant, right? It is also known as elephantiasis. It is also known as elephantiasis, right? Now next is syphilis. Syphilis is an STD, dear students, very important. Syphilis is an STD. Now STD means sexually transmitted diseases, and this syphilis is caused by a spirochete or a spiral bacteria, which is Treponema pallidum. Many a times it is asked in exam. So syphilis will be matched with Treponema pallidum, which is a spiral bacteria. Okay. Now next, ringworm. Ringworm is a fungal disease which is caused by trichophyton fungi. Gonorrhea is again an STD, and this gonorrhea is caused by Neisseria gonorrhea. Then we have pneumonia. Pneumonia is a bacterial disease that affects your lung. It is not exactly bacterial disease, but most commonly bacteria is involved in it. It could be caused by virus or fungi as well. But the most common example of the bacteria that causes pneumonia is Haemophilus influenza, and this Haemophilus influenza is a bacteria and not a virus. Please remember that it is a bacteria. Now let's see what will be the correct option. A is marked with 2, B is marked with 1, matched with 1. Okay, then 3, C is matched with 3, D is matched with 4, and E is matched with 5. So I think first will be the correct option. A2, B1, C3, D4 and E5. So correct option is 1. Now let's move on to our next question, which is related to the blood groups. Okay, human with A blood type can receive blood from. Who gave the blood grouping system? Blood grouping system was given by Landsteiner, right? So let's write this down as well. Okay, blood grouping system was given by Landsteiner, right? So according to Landsteiner, if a particular antigen is present on the surface of RBC, the corresponding antibody will be absent from the plasma. Okay, so based on this, we know that there are following blood groups: A, B, O and AB, right? Now in this question it is asked that a person with A blood type can receive blood from. So that person can receive blood from the individual who has A blood group and the individual who has O blood group. Why O? A is acceptable, right? Everybody knows that obviously if a person is having A blood group, he or she can receive blood from A blood group individual. But we should know that O is the universal donor. O blood group is the universal donor. Why? Because there are no antigens on the surface of RBC. So if there are no antigens on the surface of RBC, it cannot initiate the formation of antibodies in the recipient, right? So A and O both can donate blood to the individual with A blood group. Okay, so the correct option is second one. Now let's move on to our next question. The enzyme activator of trypsinogen. Trypsinogen is the inactive form of enzyme trypsin, and pepsinogen. Pepsinogen is again an inactive form of pepsin enzyme, are secreted respectively by. First, let's understand who activates this trypsinogen and this pepsinogen. Let's understand. So trypsinogen is converted into trypsin. Okay, first recall from where this trypsinogen is released. This trypsinogen is released from pancreas. Okay, so I will write it down so that you can remember this, right? So pancreas releases trypsinogen, and this trypsinogen is converted to trypsin by an enzyme enterokinase. Please note it down. This is a very important information. Enterokinase activates trypsinogen into trypsin, and this enterokinase is released from intestine. I will summarize all the important information related to our question, right? So this enterokinase will activate trypsinogen into trypsin. Now coming to pepsinogen. Pepsinogen, which is converted into pepsin by HCl in the stomach. Now this HCl is released from the oxyntic or parietal cell. So this is released from oxyntic or parietal cell, which is present in the lining of our stomach. Okay, so now we know the answer that trypsinogen will be activated into trypsin by the action of Krebs of Leverkusen, right? And pepsinogen will be converted into pepsin by the action of HCl that will be secreted from parietal cell. Okay, so the correct option in this question 58 is option number 3. Let's move on to our next question. Which of the following is correct with respect to cockroach? Okay. So 1. Genital pouch is bound dorsally by 10th tertium in female. Ejaculatory duct opens into male gonopore, which is ventral to anus. This is the correct option, dear students. Okay. In case of cockroach, in case of male cockroach, the ejaculatory duct opens into male gonopore. That is right. And this gonopore is ventral to anus. Okay, this is a very important line from NCERT, from which question is asked in NEET. Now, foregut and midgut are lined by cuticle. No, only foregut is lined by cuticle. Okay. Spermatophores are produced in the spermatheca. No. Spermatheca is present in female cockroach. Spermatheca is present in female cockroach and not in male cockroach. Spermatophores is made in seminal vesicle of male cockroach. So it will be formed in seminal vesicle and not in spermatheca. Spermatheca is a bag-like structure in female cockroach that will help in collecting the spermatophores at the time of copulation, right? So spermatheca is present in female cockroach. Spermatophores are produced in the seminal vesicle. Spermatophores are the packets of sperm which are produced in the seminal vesicle of male cockroach. Okay. So the correct option, correct answer for this question number 59 is 2. Here you can see very important information, the genital pouch in female. The genital pouch in female is formed by the 7th, 8th, 9th sterna. Okay, by 7th, 8th, 9th sterna, which forms the brood pouch. Similarly, in male cockroach, the genital pouch in males is surrounded by dorsally 9th and 10th tertia. Dorsal surface means the upper surface of cockroach. It is surrounded by 9th and 10th tertia, and ventrally it is surrounded by the 9th sterna. Okay, can you recall the chitinous plates? The chitin plates on the dorsal surface of cockroach are known as tertia. Okay, tertia, and ventrally it is known as sterna, and laterally it is known as pleura. And these chitin plates are joined to each other by arthrodial membrane. Remember this. This is also a very important question. Now let's move on to our next question. Read the following. Cleidoic eggs. What are cleidoic eggs? Eggs which has hard shell made up of calcium carbonate, but it can exchange gases. That means it is porous, right? So can you recall the eggs of hen that you eat? That is cleidoic egg. Scales on limbs, dry skin, 4-chambered heart, poikilotherm. The above characters are found in alligator. Who is alligator? Alligator is a reptile. So I will write it down. Alligator is a reptile. Corvus. Corvus is crow. Crow is aves. Aves means birds. Naja. Naja is the cobra, snake. So Naja is again reptile. And then platypus. Platypus is ornithorhynchus, which is egg-laying mammal. So platypus is egg-laying mammal. Right. Now let's see in which of the following organism all the features will be there. Please pay attention to poikilotherm. Everyone pay attention to poikilotherm. What do you understand by poikilotherm? The organisms that cannot regulate their internal body temperature, they are known as cold-blooded animals or poikilotherms. So poikilotherms are cold-blooded animals. We know from our knowledge of animal kingdom that mammals and birds are homeotherms. So aves and mammals are homeotherms, right? So they are eliminated. Now we have alligator and Naja. So here you can see cleidoic egg, scales on limbs. Naja does not have limb. Okay, Naja is a cobra. Cobra don't have limbs. So again this option is eliminated. Now we have only alligator. So alligator will have cleidoic eggs, will have scales on limbs, will have dry skin, 4-chambered heart, and it is again cold-blooded animal or poikilotherm. So the correct option for our question number 60 will be 1. Okay. Next question, question number 61 on your screen. Which of the following wrongly represents the nature of cardiac muscle? Pay attention to wrongly. Striated involuntary muscle, yes. Yes, heart muscles are striated and they are involuntary. Intercalated disc with gap junction, absolutely correct. This is the characteristic feature of your cardiac muscle. They have intercalated disc, okay, which is actually gap junction that will help in contraction of the cardiac muscle as a unit. It will also amplify the wave of contraction, okay. Parallel bundles of muscle fiber, no, this is wrong. The muscles of heart, cardiac muscles are branched and not parallel. So they are branched. Very important. They are not parallel. Neural regulation by autonomic nervous system, yes. It is not under our control. So it is not innervated by somatic nervous system. It is innervated by autonomic nervous system. Okay, so correct option for this question will be this one, which is wrong statement. Here you can see, this is beautiful picture of our heart with the cardiac muscles. You can see the cardiac muscles are branched, right? Striations are there, intercalated disc are there. Intercalated disc is actually gap junction. We know now what is gap junction. Gap junction is a junction which is present in the epithelial tissue, and this here it is not epithelial tissue, it is muscular tissue. It will be present between the tissue or the cells of the tissue, and this gap junction will allow the movement of small molecules from one cell to another. This gap junction is important here because it will allow all the muscles of the heart to contract as a unit, and it also amplifies the wave of contraction. These gap junctions are cytoplasmic extensions, right? So contractile tissue, cardiac muscle present only in the heart. The cell junctions formed by fusion of plasma membrane of the cells. These cells are stuck together, right? Intercalated disc are communication junction that allow them to contract as a unit, right? So this was about cardiac muscle. Now let's move on to our next question, question number 62. In a cross between a normal male and a heterozygous female for color vision, imagine the male is normal, okay, but the female is heterozygous for color blindness. Female is heterozygous for color blindness. She is a carrier. She is not affected, but she is a carrier. They are asking what percentage of the progeny will be color blind, 50%, 75%, 0% or 25%. Let's make this cross. So the progeny of the F1 generation will have one female that is normal, okay, one male who is normal, then there will be one female who will be carrier, and ultimately one male who will be normal. Okay, let's check it again. So X and XC, can you see X and this X? So this will be female who is carrier, right? Then there will be an affected male. Then there will be one carrier female, and then there will be a normal male. So let's write it down, every possibility. So here we will have affected, no, not affected, sorry, carrier female. This is carrier female. Okay, this is carrier female. This is the affected male. Remember, color blindness is X-linked recessive disorder. That means a person can be affected only if both the X chromosomes are affected, right? Now this is again a carrier female, and this is a normal male. Okay, now let's see the question again. What percentage of the progeny will be color blind? Okay, so what percentage? Out of 4 individuals, only one is affected, so the percentage is 25%. Okay, so 25% will be your correct answer. Copper releasing IUD. So what is the copper releasing IUD? Multilode 375. Copper releasing IUD IUDs are copper T, copper 7, multilode 375. And what is the action of copper? Copper will reduce the motility of sperm and ultimately it will affect the fertilizing capacity of the sperm. So what will this copper do? This copper will reduce the motility of sperm and it will reduce the fertilizing capacity of sperm. Okay, why? Because copper will interfere with the functioning of mitochondria in the sperm, and we know mitochondria will provide ATP for the motility of the sperm. So if mitochondria will not be able to function properly, the motility of the sperm will be reduced. Ultimately, the fertilizing capacity of the sperm will be reduced. Okay. LNG20 is the hormonal releasing IUD. So LNG20 and Progestasert are the hormone releasing IUD. Lipus loop is the non-medicated IUD, and Progestasert is again hormone releasing IUD. Okay. So this was about question number 63. Option 2 will be correct. Let's move on to our next question, question number 64. Choose the correct set of statements related to the given picture showing a phase of cardiac cycle. So recall, cardiac cycle is the activity of the heart in one beat. During one beat, what all activity of the heart is there, that is included in cardiac cycle. Cardiac cycle is of 0.8 seconds, right? And this cardiac cycle is broadly divided into 3 phases, that is atrial systole, ventricular systole and joint diastole, right? Atrial systole, ventricular systole and joint diastole, right? Atrial systole is for 0.1 second, ventricular systole is for 0.3 second and joint diastole is for 0.4 second, repeatedly asked in exam, right? Wow, beautiful. Now let's see what is this depicting, which phase of cardiac cycle is this image depicting. So here you can see the ventricles are contracting, that means it is showing us ventricular systole or ventricular contraction. So what happens during ventricular systole? Let's see, there are following statements, let's read them one by one. Second heart sound is heard. Increase in ventricular pressure causes closure of AV valves. Yes, absolutely correct. The atrioventricular valve, that is the bicuspid and tricuspid valve, will be closed at the time of ventricular contraction, and yes, there will be increase in the pressure inside the ventricles. Pumping of blood into aorta, yes, there will be pumping of blood into aorta as well. And will there be passive ventricular filling? No, ventricles are contracting, pressure is increasing, there could not be passive filling of the ventricles, no. So the correct option will be B and C. That means option number 2 is right. What about second heart sound? Second heart sound is due to closure of semilunar valve. Here in the later stage you will see there will be opening of semilunar valve because we want our blood during ventricular contraction to be pushed in the aorta and pulmonary artery. So semilunar valve should be closed and not open. Should be open and not closed. So this is wrong and this is wrong. Correct option for this question will be second one. B and C statements are correct. Okay, let's see in detail. Ventricular systole. Okay, ventricular systole, which is for 0.3 seconds in a cardiac cycle, is divided into two phases. One is early ventricular systole. Another one is late ventricular systole. In case of early ventricular systole, the ventricles will contract, atria will relax, atrioventricular valves are closed, AV valves are closed, semilunar valves are also closed. Okay. So ventricular contraction causes the AV valves to close. Ventricular contraction will cause the bicuspid and tricuspid valve to close. Now coming to late ventricular systole, again ventricles will contract, atria will be in the relaxed state, atrioventricular valves will be closed and semilunar valve should be open. Now the semilunar valves will open and the blood is pumped to the arteries. Which arteries? Aorta and pulmonary artery. Right? Now let's move on to our next question. Question number 65. Match list. List 1 and list 2. In list 1 we have canal system, oil gland, air bladder, poison gland and parapodia. In the list 2 we have Setacula, Catala, Scypha, Naja and Nereis. Now canal system is present in Scypha. What is Scypha? Scypha is a porifera. Okay, or sponge. It is also known as cycon. Right? And we know that our porifera or sponges have water canal system. So canal system will be marked with Scypha. Oil glands are present in aves. Oil glands which are known as preen glands. These are the only glands which are present at the base of the tail of aves. So Setacula is an ave. Setacula is parrot. Setacula is parrot. Okay. Parrot is an ave. Right. Now air bladder. Air bladder is present in bony fishes. So here bony fish is Catala. Okay, it is a bony fish or ostrich thighs. These bony fishes have air bladder inside them so that they need not to swim continuously. Okay, they can float in water because of this air bladder. Poison gland will be present in Naja. Naja is the cobra, belongs to reptilia. And parapodia is present in Nereis. Very important. What is Nereis? Nereis is an annelida. It is also known as sandworm. Okay, and this Nereis or sandworm has lateral appendages, parapodia, that helps in swimming as well as exchange of gases. Right. So now let's see what is the correct option. A will be marked with 3. B will be marked with 1, C will be marked with 2, C will be marked with 2, only one option, second one. Okay, D will be marked with 4 and E will be marked with 5. So correct option for this question is second one. Fine. Now let's move on to our next question. Choose the incorrect statement about juxtaglomerular cells of humans. Okay. Now juxtaglomerular cells. Let's read the statement. Cellular modification of afferent arteriole. Secrete enzyme renin. Secrete erythropoietin which stimulate erythropoiesis in red bone marrow. Special sensitive region of DCT. This is the wrong statement. Okay, JG cells of humans are not the special sensitive region of the DCT, but it is the cellular modification of afferent arteriole. So this is wrong. Let's understand this in detail. Juxtaglomerular cells are actually part of juxtaglomerular apparatus, right? And in juxtaglomerular apparatus, these JG cells will secrete the enzyme renin that is responsible for renin, angiotensin, aldosterone system, that is the RAS mechanism, and this RAS mechanism helps in increase in blood pressure. Important. Okay. Now let's move on to our next question. Match list 1 with list 2 and choose correct answer from the given option. So in list 1 we have evolution by anthropogenic action, then adaptive radiation, spontaneous generation and mutation. See, evolution by anthropogenic action, I will match it with DDT resistance in mosquito, isn't it? When DDT was discovered, extensively used by humans for its benefit, then what happened? Slowly the mosquitoes became resistant to DDT. Okay. Now anthropogenic means man. Okay, anthropogenic means related to the actions of man, and obviously DDT resistance in mosquito is due to anthropogenic action. Adaptive radiation, I will match it with marsupials of Australia, isn't it? In Australia, all the marsupials are developed from a single ancestor, right? So it is an example of adaptive radiation. Spontaneous generation. Spontaneous generation means living forms arising from the non-living matter spontaneously, right? That mutation. So mutation is a discontinuous variation. We cannot predict whether the changes or the adaptations will be for the good or for the benefit of the individual. No, we cannot predict. So it is a discontinuous variation, right? Now let's see what will be the correct option. A will be marked with 4, B will be marked with 1. B will be marked with 1, only one option is correct, that is the fourth one. Okay, then C will be marked with 2 and D will be marked with 3. Fine. So the correct option for question number 67 will be fourth one. Let's move on to our next question. Select the favorable condition required for dissociation of carbon dioxide from hemoglobin. Now, when will this dissociation occur? When your hemoglobin will be dissociated from the carbon dioxide? When partial pressure of oxygen is more. So, correct option here will be high PO2 will lead to this dissociation. Not low pH, not high temperature and not high PCO2, right? So correct option is whenever the PO2 will increase, there will be dissociation of hemoglobin and carbon dioxide. Okay. Let's move on to next question, question number 69. The joint between the pubic bone of the pelvic girdle is, okay. So there is a joint between the pubic bones in the pelvic girdle which is known as pubic symphysis. And this pubic symphysis is actually a cartilaginous joint. This is a cartilaginous joint. And this cartilaginous joint dissolves at the time of birth and forms a birth canal. Remember that. This will form the birth canal, right? So is it a ball and socket joint? No. Fibrous joint? No. Gliding joint? No. Correct option is cartilaginous joint for question number 69. Now next question, question number 70. Which of the following is not a step in multiple ovulation embryo transfer, that is MOET? Multiple ovulation embryo transfer. What happens in this technique? The cattle or the cow is administered with FSH like hormones, because of which there is super ovulation. That means multiple ovum will be released. Otherwise there will be release of only one ovum, but we want multiple ovum to release. So that's why we will administer the cow with FSH. So there will be release of many ovum. Then what will happen? We will artificially inseminate the cow, or we will mate that cow with an elite bull. Okay. Now all the ovums will be fertilized. Then we will let the zygote to grow into 8 or 32 cell stage. Now that embryo will be then retracted and will be placed in various surrogate mothers. So what we will get, instead of one calf, we will get 6 to 8 calves by this single process, and this is what is MOET, multiple ovulation embryo transfer. Now let's see those statements one by one. Cow is administered hormone having FSH activity. True. Cow yield about 6 to 8 eggs at a time. Yes, multiple ovulation. The zygotes are transferred to the uterus of surrogate mother. No, the embryo is transferred. So the embryo with 8 to 32 cell stage is transferred and not the zygote. So this is wrong. This is wrong. Cow is either mated with an elite bull or artificially inseminated. Absolutely correct. Okay. So the correct option for this question number 70, because they have asked which is not a step of MOET. So your answer will be the third one. Okay dear students, hope you all are enjoying this session. Please don't forget to like this session, share it with your friends and subscribe Infinite Learn NEET channel. Okay. Now select the hormones which raises during the proliferative phase of menstrual cycle. Remember, the proliferative phase is also known as the follicular phase. Okay. So proliferative phase is also the follicular phase. Now in this phase there is increase in the level of FSH, there is increase in the level of LH, and there is increase in the level of estrogen. Okay. FSH and LH are released from the anterior pituitary of the female under the influence of gonadotropin released from hypothalamus, and estrogen will increase because of release of estrogen from the follicular cells. The growing follicular cells will release estrogen, not progesterone. So correct option here will be estrogen, FSH and LH. Gonadotropin will increase, yes, but not progesterone. Not progesterone, because progesterone will be secreted by corpus luteum after ovulation in the luteal phase. So then the progesterone level will increase, but not at the time of follicular phase or the proliferative phase. Okay. Now let's move on to our next question, which is question number 72. Beta thalassemia differs from alpha thalassemia in this aspect. Okay. So remember, in beta thalassemia there is mutation of the chromosome number 11, but in alpha thalassemia there is mutation in the chromosome number 16. Okay. So in the chromosome number 16, HbA1, HbA2 genes are defected in case of alpha thalassemia, but in beta thalassemia there is defect in HBB gene in the chromosome number 11. So this is the correct option for question number 72. Match list 1 and list 2 and identify the correct set. You know, nowadays in NEET there are many match the following questions. Okay, so please pay attention to the accuracy. Alexander von Humboldt. Okay, so Alexander von Humboldt gave the species area relationship. Charles Darwin gave the theory of natural selection. Paul Ehrlich gave the rivet popper hypothesis. Ramesh Chandra Dagar is known for organic farming. And Tillman is related to the ecosystem experiment in the outdoor plots. Okay, if you remember rivet popper hypothesis, all the species present on the earth is you can say is similar to the rivets. Rivets means the nuts and bolts of an airplane, right? If we remove the nuts and bolts from the corner of the airplane, there will not be any problem. But if we remove the important nuts and bolts from the airplane, then it will crash. Okay, similarly, it is similar to our ecosystem. If the important species are eliminated, it will lead to crash of our ecosystem. So this is the rivet popper hypothesis given by Paul Ehrlich. Now let's see what is the correct option. A is matched with 4. Yes, everywhere it is matched with 4. Charles Darwin. B is matched with 2. Only one option is there. C is matched with 1. Yes, and D is matched with 5. And E is matched with 3. The correct option for this question number 73 is second. Now, question number 74. Choose the correct set of statements related to immunization by vaccine. It develops immunological memory. Yes. It is characterized by pathogen-specific immunity. Yes. The first response to the inactivated weakened microbe in the vaccine is of low intensity. Yes. It develops artificial, active, acquired immunity. Artificial, active, acquired immunity. Yes, this is also correct. All the statements are correct. That means correct option will be 4. Let's understand what is immunization by vaccine. Vaccine are example of artificial, active immunity. Right? Now, vaccines will have attenuated or heat killed pathogens or part of pathogen. So, what these pathogens will do is they cannot cause disease because they are less in number, right? But they can activate our immune system to form antibodies against them, right? So, we know that first response will take time. So, first response will be slow. But next time when our body will encounter the same pathogen, now our body is ready to make antibodies to fight that pathogen, and that is the secondary response. Secondary response is strong and quick. Okay, so correct option for this question number 74 is 4th one. Let's move on to our question number 75. Test Tube Baby program is an assisted reproductive technology which includes in vitro fertilization and embryo transfer, in vivo fertilization and intrauterine transfer, artificial insemination and embryo transfer, gamete intrafallopian transfer and embryo transfer. Test Tube Baby program is assisted reproductive technology, that is ART. In this, the fertilization is taking place in the test tube outside the body, and this is known as in vitro fertilization. This is in vitro fertilization, fertilization in a test tube, and then the embryo is transferred either to the mother or the surrogate mother, right? So correct option for question number 75 is first one. In vivo fertilization means fertilization inside the body, that is not happening in test tube baby. Artificial insemination is the introduction of sperm with the help of tube in the female reproductive tract. So this is not the test tube baby program. Then gamete intrafallopian transfer, this is known as gift, in which the gametes are transferred into fallopian tube and allowed to fertilize, and that is in vivo fertilization and not in vitro fertilization. So correct option for question number 75 is 1. Next, statement 1 and 2. Automobiles equipped with catalytic converter should use unleaded petrol. This is right, yes. Lead in petrol activates the catalyst? No, it deactivates the catalyst. Right? That's why it is said not to use leaded petrol that contains lead, because it will deactivate the catalyst, and now our catalyst will not be able to convert the gases into carbon dioxide and water. So statement 1 is correct and statement 2 is incorrect. That means the correct option or correct answer for this question number 76 is third one. Next question. Question number 77. Early diagnosis of disease is possible based on the principle of antigen-antibody interaction. So antigen-antibody interaction will cause agglutination, and this is seen in ELISA. So yes, the correct option here will be ELISA. ELISA is enzyme linked immunosorbent assay, and we know that ELISA is used for detection of HIV, okay, in the body. Okay, Western blot is for detection of protein. Southern blotting is related to DNA. Okay, and autoradiography is again related to detection of the DNA that is hybridized with DNA probe, radio labeled DNA probe, right? So the correct option here, where antigen antibody reaction will occur, that is ELISA. Okay, so ELISA is based on the principle of antigen antibody reaction. By this we can detect the infection of pathogen, antibodies against the pathogen. Types of ELISA is direct, indirect and sandwich ELISA. Okay, now let's move on to our next question. Question number 78. List 1 and list 2 are given. Let's match it again. Pons. So in our hindbrain we have pons, medulla and cerebellum. Pons will have pneumotaxic center. Now this pneumotaxic center is related to stopping of breathing, right? Not exactly breathing, stopping of inhalation or inspiration. Hypothalamus is related to thermoregulation. Very important question for NEET. Cerebellum is the gyroscope of our body. Gyroscope means it helps in balancing. Okay. Limbic system is related to our emotion. So limbic system is also known as emotional brain. Occipital lobe helps in vision. So I will match it with vision center. So what will be the correct option? A with 2, B with 1, C with 4, C with 4, 2 options, D with 5, D with 5 again 2 option, E with 3, E with 3 again 2 option. Let's see, I will have to check it. So A with 2, A with 2, 2 options. No, everyone has A with 2. Hypothalamus, B with 1, B with 1 only 2 options, 2 and 3. Cerebellum, 3, C with 4, C with 4, yes, third is the correct answer. D with 5, D with 5, yes. Occipital lobe, E with 3, yes, absolutely correct. So the correct option for 78 question number is third one. Okay. Now let's move on to 79 question. Biological response modifiers which help in destroying tumor by activating natural killer cells are alpha-1 antitrypsin, no. Alpha globulin, alpha lactalbumin, correct option is alpha interferons. So interferons are used in the treatment of cancer. Okay, why? Because these alpha interferons activate the natural killer cells which go and destroy the tumor cells. So alpha interferons will activate the natural killer cells, it will go and kill the tumor cells. So that's why it is used in treatment of cancer. For question number 79, the correct option will be 4. 18. Choose the answer with correct description of the following cells. Beta lymphocyte, surface antibody, cell mediated immunity. Thrombocytes are cell fragments which causes blood coagulation, absolutely correct. Thrombocytes are platelets, these are not actually cells, these are only cell fragments. Osteocytes are enclosed in lacuna, that is right, but they don't help in bone resorption. For bone resorption we have osteoclast. Osteoclast are the cells that help in bone resorption. They are multinucleated and they are present in a bony depression known as Hauschup's lacuna. Okay, so osteoclast will help in bone resorption. Brush border cells, microvilli help in defense? No, it will help in increasing the surface area for absorption. So the correct option for this question number 80 will be the second one. Let's move on to our next question. An increase in blood fluid volume can suppress ADS secretion by hypothalamus? Yes. So if blood volume is increasing, okay, we don't want our body to reabsorb water from DCTN collecting duct. Ultimately, what is the function of ADH? This ADH will cause reabsorption of water from DCTN collecting duct, blood volume will increase. Already if blood volume is increasing, the ADH activity will be suppressed, this is correct. Osmoreceptors are located in neurohypophysis? No. Osmoreceptors are located in hypothalamus. Right? So one is correct, second one is incorrect. Statement 1 is correct, statement 2 is incorrect. Correct option for this question will be 3. Understood? Next question. Read the following: multiple allelism, codominance, dominance, polygenic trait, incomplete dominance. Which of the above are related to blood groups of human? So how many blood groups we have? A, AB given by Landsteiner, O and B. Right? So these are example of multiple allelism, yes. These are example of codominance. These are example of dominance as well, but not polygenic trait and not incomplete dominance. So A, B and C are correct. So correct option for this question will be option number 2. Next question. Nowadays ozone thickness is decreasing in stratosphere due to this reason. Why our ozone layer is getting thinner and thinner? Because of the chloride atoms formed from chlorofluorocarbon due to UV rays degrades ozone. Right? Now this chlorofluorocarbon is used in our refrigerators for refrigeration. So it will release free radicals of chloride and ultimately it will react with our ozone layer, okay, and it will be degraded, right? So correct option will be option number 4. Next question. Extinct animal of Africa is quagga. Quagga is extinct from Africa, not thylacine, not dodo, not stellar sea cow. So correct option for option number 84 is 1. Now let's move on to our next question, question number 85. Which of the following center is not found in medulla oblongata? Not. Every time you see this not, incorrect, correct word, please mark it like I am marking here. So pneumotaxic center, just now we have discussed, it is present in pons and not in medulla. So this is the not answer here. Respiratory rhythm center is in medulla. Regulation center for heart rate is in medulla. And vomiting center is again in medulla. So what is not? Pneumotaxic center, because it is in pons, right? Chalo, next question. In the absence of superior competing species, the inferior species has greater distributional range. This explains. Okay. So, if in a classroom there are various students and if the topper is absent in a particular exam, obviously the second topper will be the topper, isn't it? So, the inferior species has greater distributional range in absence of the superior competing species. This is Cornell's competitive release principle. So, correct option for this question is first one. Okay. Which of the following statement is not correct? Pyramid of biomass in forest is inverted. Pyramid of number in ocean is upright. Pyramid of energy is always upright. Pyramid of biomass in sea is inverted. Okay. So, let's understand with this picture. Can you see? Okay. So, pyramid of number, pyramid of biomass, pyramid of energy. There can be three pyramids. And pyramids of biomass in an aquatic ecosystem is inverted. So, here, what was the statement? Which is not correct. So, pyramid of biomass in the sea is inverted. This is correct. Pyramid of energy is always upright. This is also correct. Pyramid of number in the ocean is upright, and pyramid of biomass in forest is inverted. Only two options we need to find out. Let's see. Okay. So, we know that pyramid of biomass in forest, biomass, what do you understand by biomass? It is the organic matter which is present in the individual, right? So, pyramid of biomass in a forest and the pyramid of number in a ocean is upright. This is also true. The incorrect statement is this one. Okay, the biomass of forest should be upright and not inverted. Okay. Now, here you can see in the image also, pyramid of number is upright, pyramid of biomass can be upright or inverted, and pyramid of energy will always be upright. Remember this point. Okay. Now, let's move on to our next question, question number 88. Venereal disease which spreads from infected mother to fetus. Venereal diseases are the STDs or the sexually transmitted diseases. Okay. So, smallpox, HIV, malaria and diarrhea, out of this only HIV is the venereal disease or STD which is passed on from the mother to the developing fetus. Okay. So, correct option for question number 88 is 2. Match the following: Hirudinaria, Pleurobrachia, Acidia, Ornithorhynchus and Ascaris. So, Hirudinaria is an ectoparasite. What is Hirudinaria? Hirudinaria is blood sucking leech. Okay. Hirudinaria is blood sucking leech. It releases an anticoagulant, hirudin. So, what is hirudin? It is an anticoagulant. Now, this anticoagulant will keep the blood from coagulating and this leech can suck your blood, right? Pleurobrachia has the property of bioluminescence. Pleurobrachia is a ctenophora. Acidia, Herdmania, Acidia, Doliolum, Salpa, these are the examples of Urochordata, and Urochordata shows retrogressive metamorphosis. That is, we always see that larva is less developed and the adult is developed, but in case of Urochordata, the larva is more developed with notochord in the tail region है ना, and it is moving as well, but the adult is less developed, so it is known as retrogressive metamorphosis, right? Ornithorhynchus is our very own platypus. This platypus is egg-laying mammal. Okay, so I will connect it with connecting link, okay, as it is the connecting link between the mammals and the reptiles, right? Then we have Ascaris here. Ascaris or roundworm is an intestinal parasite. Now let's see what is the correct option. A will be marked with 4, B will be marked with 2, C will be marked with 5, D will be marked with 3, E will be marked with 1. So correct option for this question is option number 2. Okay. Now next question. Pyloric sphincter guards the opening between. So if this is the stomach is a J shape. No, this is not stomach, wait. So this is the stomach. I will make it, wait, okay. Now this is the stomach. Now stomach is divided into fundus, cardiac part, body and pylorus. So this is the body of your stomach. This is the cardia, which is near to heart. This is the fundus and this is the pylorus. Okay. Now the question is asking about pyloric sphincter. So pyloric sphincter will connect the stomach to duodenum, right? So here you will find the pyloric sphincter. Okay. There is one more sphincter here which is known as gastroesophageal sphincter or the cardiac sphincter. Now this cardiac sphincter will prevent the regurgitation of the content of the stomach. Our stomach is doing churning movement. It is mixing the partially digested food with the gastric juice. We don't want this content to go up. So that's why we have cardiac sphincter here. But for question number 90, the correct option is fourth one. Next question. Parathormone raises the blood calcium level by promoting the bone resorption, that is correct. Promoting reabsorption of calcium in the renal tubules, that is also correct. Leading to an increase in the calcium absorption in the GI tract, that is also correct. I think the question should be parathormone raises the blood calcium level except, okay, how it increases the blood calcium level. They are not asking that. They are asking by which method it cannot accept or cannot increase the blood calcium level. Then your answer will be increase in BMR, because BMR has nothing to do with the parathormone, right? And all this will help in increasing the blood calcium level with the help of parathormone, right? Okay. Now, you know now that calcitonin is the hormone which is antagonist to parathormone, right? So, calcitonin reduces the blood calcium level and parathormone increases the blood calcium level. Which of the following is not an important component of parturition? We know what is parturition. Parturition is childbirth. It is childbirth. Now we have 4 options: increase in estrogen and progesterone ratio, synthesis of prostaglandin, secretion of oxytocin and cortisol, secretion of prolactin. Secretion of prolactin has nothing to do with parturition or childbirth. There should be increase in the estrogen and progesterone ratio. There should be increase in the prostaglandin. Now what is prostaglandin? Prostaglandin is a derived lipid and it helps in smooth muscle contraction. It helps in smooth muscle contraction, right? Now, prolactin is the hormone that helps in formation of milk in the mammary gland. It helps in formation of milk. In the mammary gland, and you should know oxytocin is the one that help in ejection of milk. That's why it is also known as milk ejecting hormone. Okay, so correct option here was option number 4. 93. Phenylketonuria is not a chromosomal disorder, a recessive Mendelian disorder, an autosomal disorder, an inborn error of metabolism. Phenylketonuria is not a chromosomal disorder. Now question number 94. Read the following: pupil constriction, piloerection, increase in breathing rate, hypoglycemia, interacts with the intracellular receptor of the target cell. Which of the above effects are caused by norepinephrine? Norepinephrine is also known as noradrenaline. Noradrenaline. Okay, and this noradrenaline will cause pupil dilation and not constriction. Piloerection will be there. Okay, goosebumps that you get, that is piloerection. Increase in breathing rate, yes. Not hypoglycemia because it will cause hyperglycemia. Interact with intracellular receptor of the target cell, no. Okay, so correct option for this question will be only B and C. B and C, yes. So first is the correct option. Now why don't it will react with the intracellular receptor of the target cell? Because norepinephrine cannot go inside the cell. Okay, it will bind to the receptors present on the plasma membrane, which are known as surface receptors, and then there will be formation of secondary messengers, right? So the correct option here is B and C only. Question number 95. Match list 1 and list 2 and choose correct answer. Graves' disease. Graves' disease is characterized by bulging of eyeball. It is also known as exophthalmic goiter. So Graves' disease is also known as exophthalmic goiter. Okay, this is a condition of hyperthyroidism. Exophthalmos. Exoph means outside, ophthalmos is related to your eyes, so your eyes will appear like it is bulging. There will be increase in blood pressure, there will be reduction in weight. Okay, so Graves' disease I will connect it with hyperthyroidism. Rheumatoid arthritis is an autoimmune disorder that will cause inflammation of joints. Alzheimer's disease will be related to dementia, that is memory loss, and this is due to reduction in acetylcholine enzyme. Acetylcholine neurotransmitter, not enzyme. Okay. Bright's disease is related to inflammation of glomeruli, and diabetes insipidus is related to hyposecretion of vasopressin or ADH. We know this ADH or antidiuretic hormone is released from the posterior pituitary. Now this ADH is formed in hypothalamus but stored in posterior pituitary and released from there. Right? Now let's see what will be the correct option. Graves' disease, A will be marked with 3. A with 3, B with 4, C with 5, 1 will be the correct answer. D with 1, E with 2, D with 1, E with 2. Yes, so this is the correct answer. Remember, diabetes insipidus, I not I. So insipidus is not related to deficiency of insulin. Diabetes mellitus is related to deficiency of insulin. Remember this point, very important. Next question. When the calcium level in the sarcoplasm raises. So in the sarcoplasm is if the concentration of calcium ions will increase, what will happen? The active site for myosin are masked? No. See what will happen when calcium ions will increase. These calcium ions will bind to troponin C. Troponin C, and when it will bind to troponin C, it will pull tropomyosin. It will pull tropomyosin, because of which the myosin binding site on actin will be exposed. Let's write it down. Because of which the myosin binding site on actin is exposed. So let's see what is the correct option for this question. It leads to binding of calcium with a subunit of troponin on actin filament. Absolutely correct. Nerve fiber releases acetylcholine due to depolarization. No. Thick filament slide over thin filament into edge zone. No. Correct option for this question is 2. I told you why. Okay. Next, assertion and reason type of question. Natural selection enables better survival of animals with adaptability. Yes, this is right. Okay, all those adaptations which are favorable. Okay, so organisms that have those type of adaptation will survive. Adaptability has no genetic basis. Yes, there is genetic basis. Adaptability may be related to the genetic changes. So this is wrong. Assertion is correct, but reason is false. Correct option will be 3. Choose incorrect pair. Biodiversity hotspots. Species richness and endemism, yes. Acceto conservation, wildlife safari parks, yes. Sacred groves of Meghalaya, last refuges for rare and threatened plants, yes. Which is Khasi and Jaintia hills, right? Keolado National Park, Gujarat, no. It is in, tell me, Rajasthan. Okay, it is in Rajasthan and not Gujarat. Okay, so this is the incorrect pair for question number 98. The diameter of pupil of human is regulated by iris. Okay, iris are the muscles that give color to your eye. Okay, and it regulates the pupil. It regulates the pupil. Pupil is the aperture from which the light will enter and will cross your lens, right? So pupil, how much light will enter, is pupil is regulated because of the iris muscles. And remember, if the light is low or you are standing in a dim light, then what will happen? This pupil will dilate to allow more light rays to enter your eye. And if you are in a bright light, what will happen? This pupil will constrict. Okay. So correct option for question number 99 is second one. Last question. The SCN that act as a master clock is located in. So SCN that is acting as a master clock is located in the hypothalamus. So the correct option for this question number 100 is 2, hypothalamus. Okay. So that was all for today, dear students. We have solved 50 questions of zoology. I have enjoyed this session. Hope you have also enjoyed this session. Don't forget to catch us live on 17th of July after your NEET examination, because we will give you the answer keys and the solution of the NEET exam paper. Okay, that was all for today. Goodbye. Take care. See you in the next class.
Hello everyone, welcome to Infinity Learn by Sri Chaitanya. My name is Vibhati, and in today's session I'll discuss AITS Test 2, and in which I'll discuss physics portion with you. So let's start with question number 1 of physics. A physical quantity Q is related to 4 observables x, y, z and t by the relations. Q is equals to x to the power 2 by 5, z to the power 3 divided by y, t to the power 1 by 2. The percentage errors in the measurement of x is given as 2.5%. Percentage error in the measurement of y is given as 2%. Percentage error in the measurement of z is given as 0.5%. and percentage error in the measurement of T is given as 1%. The percentage error in Q will be, so percentage error in Q will be what? 2 by 5 times percentage error in X, that is 2.5, plus percentage error in Z into 3, so this is 3 times percentage error in Z, which is 0.5, plus 1 times percentage error in Y, which is 1 into 2%, plus 1 by 2 times percentage error in T, which is 1 by 2 into 1%. So this will give you percentage error in the measurement of Q as 5%, which is option 1. Next is question number 2. If the combination of the following gates, the output Y can be written in terms of inputs A and B as. So this is our AND gate, this is AND gate, and this is OR gate. So this is input A, first input of AND gate is A, B is connected with one NOT gate first. So output of this NOT gate will be second input of AND gate and it will be B bar. So this output of this AND gate 1 will be A dot B bar. Similarly, for second AND gate, first input is A, but it is going through NOT gate first. So this will be A bar and second input is B directly, and output of AND gate will be A bar dot B. Now A dot B bar and A bar dot B are the inputs of OR gate. So net output of OR gate will be A dot B bar plus A bar dot B. So correct answer is option 2. Next is 103. A long solenoid has 200 turns per centimeter. We have one solenoid, noid 1, and it has 200 turns per centimeter. That means 200 divided by 0.01 meter turns, right? So it will be 200 into 100 turns per meter and carries a current I. The magnetic field at its center is 6.28 into 10 power minus 2 Weber per meter square. So for solenoid 1, magnetic field is 6.28 into 10 power minus 2 Weber per meter square. For a solenoid, magnetic field is given as mu naught Ni. So this will be mu naught N, number of turns per unit length is 200 into 100 and I is I. Another long solenoid has 100 turns per centimeter, right? So for solenoid 2, number of turns are 100 per centimeter. We need to find it in terms of per meter. So this will be 100 divided by 1 upon 100 turns per meter. So this will be 100 into 100 turns per meter. So for second solenoid, current is I by 3 and we need to find the value of magnetic field at its center. So for second solenoid, let's say it is B and it is mu naught, N is 100 into 100 into I by 3. Let's divide this 2. So we'll get mu naught cancel, 20 cancel, 100 cancel, I cancel. So we have 6.28 into 10 power minus 3 divided by B is equal to 6. After solving this one, you'll get the value of magnetic field over here as, let's solve it now, let's solve it. This is 6.28 into 10 power minus 2. So B will be 6.28 into 10 power minus 2 divided by 6. So it will give you 1.05 into 10 power minus 2 Weber per meter square. So correct answer is option 2. Next is question 104. Match list 1 with 2. In list 1 we have electromagnetic wave type and in 2 we have its association or application, and select the correct option from the choices given below. So for list 2 we have list 1, first option is infrared lamps. So these are used for physical therapy. Second is radio waves. And out of this, 3 options which are left, it is used for broadcasting. X-rays are used to detect the fractures of bones, and ultraviolet lamps are used to kill germs in the water purifier, right? So basically A is matched with 1, B is matched with 2, C is matched with 3, and D is matched with 4. So correct answer is option 2. Question number 105. In the series LCR circuit, the voltmeter and ammeter readings are respectively. So guys, look at it. This is 200, this is 200. So potential difference across inductor and capacitor is same. VL is equals to VC. That means series LCR circuit is in resonance. It is in resonance. So effective impedance of the circuit is equal to resistance, and that is equal to 50 ohm. Ammeter and voltmeter. Ammeter reading will be, the total potential drop will be across the resistor, and this will give you RMS reading. So this is RMS value. So this will be 100 volt. Entire drop will be across 100 resistor over here, that is equal to 100 volt. So we have voltmeter reading over here, that is 100 volt. Now current will be, current reading of ammeter will be RMS current, and that is equal to V upon R, and R is 50. So this is equal to 2 ampere. So reading of ammeter is 2 ampere, whereas the reading of voltmeter is 100 volt, and correct answer is option 4. Next question is 106. The primary winding of a transformer has turns 500 and its secondary has 5000 turns. So N1 is 500 and N2 is 5000. The primary is connected to an AC supply of 20 volts and 50 hertz. So V1 is 20 volt and we need to find V2. We need to find the output through secondary coil. So frequency will not change. So frequency will be 50 hertz. So option 1 and 2 are wrong. Let's see which option is correct out of 3 and 4. So we can apply this relation N1 upon N2 is equal to V1 upon V2, right? N1 is 500 divided by N2 is 5000 is equal to V1, that is 20 divided by V2. So V2 will be, you can cancel this 2 zeros, this will be 10. So V2 will be 20 into 10, that is 200 volts. So correct answer is option 4, 200 volts and 50 hertz. Next is question number 107. In the following circuit, find I1 and I2. So this is positive terminal, this is negative terminal of the battery. So this region is at higher potential, this region is at lower potential over here. So you can see that this is PN junction, this is PN junction. P is connected to high potential, N is connected to low potential. P is connected to high potential, N is connected to low potential. So these two are in forward bias. When these two are in forward bias, they offer zero resistance, right? Ideally, they offer zero resistance, right? So they will behave like a conducting wire like this. So what will happen is current flow will be something like this. Current I will flow over here and it will follow least resistance path like this. So I1 will be completely zero. Current will not pass through this or this. It will directly pass through this circuit, this branch like this, because it is offering zero resistance. Right? So I1 is 0, so either option 1 or 4 are correct. Now what is the effective resistance? Because throughout this path, throughout the circuit, the resistance faced by the current is 2 kilo ohm, right? So I will be V upon R and V is 10, R is 2 kilo, that is 10 power 3, so this is 5 milliampere. So I2 over here will be 5 milliampere. So current I1 is 0 and I2 is 5 milliampere. So correct answer is 4. Next is question 108. If an observer is approaching a source at rest with velocity 1/5 of the velocity of the sound. So we have observer and it is approaching a source over here. Source is at rest and observer is approaching source at a velocity of 1/5 times the velocity of sound. So let's say velocity of sound is v, so velocity of the observer is v upon 5. The percentage change in the frequency of sound received by him. So let's say original frequency of sound is f and apparent frequency of sound heard by the observer is f dash. So it is given as when source is at rest and observer is moving, approaching source, then it is given as f into v plus vo divided by v, where vo is the observer's velocity and v is the speed of sound over here. So f dash will be f into v plus v by 5 divided by v. So this is f into 6v by 5v. V cancel, so we have apparent frequency as 6f upon 5. Now we need to find percentage change. So percentage change will be f dash minus f divided by f into 100%. So this will be f dash is 6f upon 5 minus f divided by f into 100. So this will be, see, this is 6 upon 5 minus 1 into f. So this is 1 upon 5 into f divided by f into 100. Ff cancel, so we have 100 upon 5 and that will give you 20%. So percentage change in the frequency received by the observer is 20%, which is option 1. Question number 109, a parallel plate capacitor is charged to a potential difference of V volts. After disconnecting the charging battery, the distance between the plates of the capacitor is increased using an insulating handle. So initially this is the situation, we are charging the capacitor with some potential difference V volt. Right, now when it is charged, we are removing this battery, so charge is fixed. Charge cannot move anywhere on the plates of this capacitor because there is no path for it to flow. Right, now after this condition, what we are doing, we are using one insulating handle to increase the separation between the plates of the capacitor, but charge on the capacitor will be same. We are increasing the separation on between the plates of the capacitor. So what will happen to capacitor? D dash is greater than D. If initial capacitor is epsilon naught A upon D, then by increasing the value of D, new capacitor will be epsilon naught A upon D dash. You can see that capacitor is inversely proportional to D over here. So if you are increasing the value of D, capacitor decreases. So new capacitor is less than the previous one. Now charge should be constant. We know that Q is equals to CV. Charge is constant before and after increasing the separation. So if Q is constant and C is decreasing, then V should increase to maintain the C into V product constant, right? So potential difference between the plates will increase. Correct answer is option 3. Question number 110. A large plane sheet of charge has surface charge density sigma. The electric field strength due to it at a distance d from it is. So we have large plane sheet and it has surface charge density sigma. We need to find electric field due to this plate at a distance d from the plane sheet of charge. So it is direct formula, which is sigma upon 2 epsilon naught. So correct answer is option 3. Question number 111, again it is direct formula based. If you remember it, you can directly mark it, right? If theta 1 and theta 2 be the apparent angles of dip observed in two vertical planes at right angles to each other, then the true angle of dip theta is given as. So we have relation between this, that is cot square theta is equal to cot square theta 1 plus cot square theta 2. So correct answer is option 4. Next is 112. Assertion is given as a real gas behaves like ideal gas at low pressure and high temperature. It is true. Reason R is at low pressure and high temperature the molecules are far apart, which is also true, and molecular interactions are negligible without interaction, the gas behaves like an ideal gas, which is also true. So assertion is true as well as reason is true, and reason is the correct explanation of assertion. So correct answer is option 1. Next is 113. Angle of prism is 30 degree. So we have this angle of prism as 30 degree and its refractive index is √2. Okay, it has refractive index of √2 and one of the surface is silvered. Let's say we are silvering this surface. So this surface will behave like a mirror. At what angle of incidence a ray should be incident on one surface so that after reflection from the silvered surface, it retraces its path? So here we are going to use that concept now. For a mirror, if the incidence is normal, then ray will retrace its path, right? So after refraction through this surface, ray should strike this surface perpendicular to it. So let's say this is the incidence ray, this is normal, this is angle of incidence I, and after refraction it should strike this surface perpendicularly with a 90 degrees so that it can retrace its path something like this, normal incidence, right? So for this small triangle, this is 30 degree, this is 90 degree, so this is 60 degree. Now again this is perpendicular, so if this is 60 degree, then this remaining angle is 30 degree because this is A dash is what? A normal to the surface of this. So this angle is 60, so remaining will be 30 because total is 90. So for this surface, I have angle of incidence I, angle of refraction 30 degree, right, and refractive index of the second medium is √2 and refractive index of the first medium is 1. So let's apply second law of refraction, that is mu1 sin I is equal to mu2 sin R. Mu1 is 1, sin I will be sin I, mu2 is √2 and sin R. So it will be sin 30 degree. It will be sin 30 degree. So this is √2 into sin 30 degrees 1 by 2. So this is 1 upon √2. So sin I is coming out as 1 upon √2. That means I should be 45 degree, which is option 3. I should be 45 degree, which is option 3. I hope everything is clear so far. If you have any doubt, just revise everything. Okay, if you have any doubt in the concept, just go and revise the concept first, then again attempt the question. Next is question number 114. Statement A, astigmatism is corrected by using cylindrical lenses. Yes, we correct astigmatism using cylindrical lenses. At sunset or sunrise, the sun looks reddish. This is also true. At sunset and sunrise, sun looks reddish because red has the maximum wavelength and low wavelength lights are scattered by the atmospheric particles, right? So over here, the only wavelength that will reach to us will be red color wavelength, right? So statement B is also true. So both A and B are true. So correct answer is option A, 1. Next is question number 115. An unpolarized light of intensity I0 is incident on a surface of a block of glass at Brewster's angle. So in this case, which one of the following statement is true? So some part of the light will be transmitted and some part of the light will be reflected. So reflected light is completely polarized when it is striking the glass at Brewster's angle, whereas transmitted light is partially polarized. Right, so reflected light is completely polarized and its intensity is less than I0 by 2. So correct answer is option 3. Question number 116. In Young's double experiment, the ratio of intensity at maximum to that of minimum is 25 is to 16. So I max upon I minimum is 25 upon 16, and it is also given as A1 plus A2 whole square divided by A1 minus A2 whole square. Because maximum intensity will be at constructive interference, so net amplitude will be A1 plus A2 whole square, and minimum will be at destructive interference, so it will be A1 minus A2 ka whole square, right? So find the ratio of amplitudes of the wave forming the interference pattern. We need to find A1 to A2. Intensity is directly proportional to A square. For maximum it will be A1 plus A2 whole square. For minimum it will be A1 minus A2 whole square. So let's solve this 2 only. Let's take a square root. So if you take the square root on both the sides, you will get under root of 25 upon 16 as 5 upon 4, and that is equal to A1 plus A2 divided by A1 minus A2. Right, so this will be cross multiplied. So 5A1 minus 5A2 is equal to 4A1 plus 4A2. So 5 minus 4A1 is A1, and over here 4A2 plus 5A2 will be 9A2. So A1 upon A2 will be 9 upon 1, which is option 2. A1 upon A2 is 9:1. Next is 117, assertion. Bohr's model is applicable only to hydrogenic, that is single electron atoms, which is true. It cannot be extended to even 2 electron atoms such as helium. It is true. Second is the de Broglie hypothesis of matter waves supports the Bohr's concept of stationary orbits. We need to find over here which option is correct. So both of the statements are correct. Bohr's model is applicable for single electron atoms, hydrogen like atoms, right? So assertion is true. The de Broglie hypothesis of matter waves supports the Bohr's concept of stationary orbits. So reason is also true, but it is not the correct explanation of assertion. So correct answer is option 2. Question number 118, half-life of a radioactive substance is 4 years. Time taken for the number of radioactive nuclei to become 12.5% of the original is. So let's say original sample is 100%. So after 4 years, it will reduce to 50%, because half-life is the time in which the sample will reduce to 50% of its initial value. Now we are left with 50%. Now again after 4 years, when you look at the sample, you'll get half of 50%, that is 25%. Again after 4 years, you'll get half of 25% only left with you, that is 12.5%. So in order to obtain 100 to 12.5% material, you need to wait for 4 plus 4 plus 4, that is 12 years, right? So time taken by this sample to become 12.5% of the original is 12 years, which is option 2. Next is 119, two projectiles of same mass and with the same velocity are thrown at an angle of 60 degree and 30 degree with the horizontal, then which will remain same? So if you add these two angles, you will get the total addition is 90 degree, right? So these two angles are complementary angles, and we know that for complementary angles the range, horizontal range is same. So correct answer is option 2, range of projectile is same. 120, when photons of wavelength lambda are incident on an isolated sphere suspended by an isolated thread, the corresponding stopping potential is found to be V. When photons of wavelength lambda by 2 are used, the corresponding stopping potential is thrice the above value, matlab it is 3V. If the light of wavelength lambda by 3 is used, then calculate the stopping potential for this case. So for the first case, we can use this equation. For the first case, EV naught is equal to HC by lambda minus phi. Phi is the work function. So over here for the first case, it is EV is equals to HC by lambda minus phi. For the second case, it is 8 times 3 times 3V into HC upon lambda. Lambda is lambda by 2 now, minus phi. Work function will not change, right? So this is 3EV is equals to 2HC upon lambda minus phi. Put the value of EV from this equation to this equation. So you'll get 3 times EV. HC upon lambda minus 3, okay, minus phi is equal to 2HC upon lambda minus phi. So this will give you 3HC upon lambda minus 3 phi is equal to 2HC upon lambda minus phi. So we'll get 3HC upon lambda minus 2HC upon lambda is HC upon lambda, and that is equal to 2 phi. So phi for u is hc upon 2 lambda. This is phi, work function. Now let's apply the same equation for the third condition. So for the third condition, I'll have to make some space here. Okay. So for the third condition, E V naught, I don't know what is the value of V naught that we need to find, is equals to hc upon lambda by 3 minus phi, which is hc upon 2 lambda. So this is 3 hc upon lambda minus hc upon 2 lambda. So this will give you what? This will be 2 lambda LCM. This will be 6 hc minus hc. So this is 5 hc upon 2 lambda. This is EV naught. I am writing it here. EV naught is 5 hc upon 2 lambda. So V naught will be 5 hc upon 2e lambda. We need to find V naught. So it is 5 hc upon 2e lambda, which is option 3. Next is question number 121. When bulk pieces of conductors are subjected to changing magnetic flux, induced currents are produced in them. Magnetic flux is changing, that is why current will induce. Their flow pattern resembles swirling eddies in water, and these currents are called eddy currents. True. The motion of a copper plate is damped when it is allowed to oscillate between the magnetic pole pieces. Because it is oscillating between the magnetic pole pieces, what will happen? Its surface will be having eddy currents, and because of that its motion will be damped, right? So both A and B are true. So let's see which option is correct. Option 3 is correct. Both A and B are true. Question number 122. A conducting circular loop is placed in a uniform magnetic field 0.025 Tesla with its plane perpendicular to the loop. Okay. This is magnetic field direction. This is area vector direction. This is the initial situation. The radius of the loop is made to shrink at a constant rate of 1 millimeter per second. dr by dt, the rate at which radius is decreasing, is 10 power minus 3 meter per second. I am converting this millimeter into meter. The induced EMF when the radius is 2 centimeter is. So flux is BA cos theta. Now theta is the angle between area vector and B vector, which is 0 degree. So it will be just BA, and it is B pi r square. Induced EMF is minus d phi by dt. So it will be minus of B pi into r square ka differentiation. So this is 2r dr by dt. And dr by dt is decreasing. So it is 10 power minus 3 with minus sign. So this will be minus B, which is 0.025 into pi into 2 into r. R is 2 centimeter. So this is 2 upon 100 into dr by dt is minus of. So this minus will cancel this minus, 10 power minus 3. So we have over here 1, 2, 3. So this is 25 into 250 into 200. So we have pi into 10 power minus 6 volt. So this is pi microvolt. Pi microvolt. So correct answer is option 2. After calculating this one, you will get pi microvolt. Next is question number 123. The velocity v of a particle at time t is given by at plus b upon t plus c, where a and b are constants. The dimensions of a, b and c are. So in this we'll have to use principle of homogeneity. The dimension of v should be equal to dimension of at, and dimension of b upon t plus c should be equal to dimension of v, because you can add like quantities and you can equate like quantities, right? So dimension of v is lt minus 1, and that is equal to dimension of a into dimension of t is t. So dimension of a will be lt minus 1 divided by t. So this is lt minus 2. Now you can add time with time. That means dimension of c should be of time. So dimension of c is equal to dimension of time, that is t. Now dimension of B upon T plus C should be dimension of V. So dimension of B is equal to dimension of V, that is LT minus 1, into dimension of T plus C is T. So this will cancel this. So we have dimension of B as L. So dimension of A is LT minus 2, dimension of B is L, and dimension of C is T. So correct answer is option 2. Next is question number 124. If m, e, tau and m respectively represents free electron density, charge, relaxation time and mass of the electron, then the resistance of a wire of length L and area of cross section A will be. So rho, resistivity in terms of relaxation time, m, e and n is given as m upon ne square tau. So resistance will be rho L upon A. So this is rho is m upon ne square tau into L upon A. So resistance is ml divided by ne square tau A. So that is equal to option 1. Next is question number 125. The given Wheatstone bridge is showing no deflection. That means this Wheatstone bridge is balanced. In the galvanometer joint between the points A and B, the values of resistance R as shown in the circuit is. So basically if this is P, this is Q, this is R and this is S, then we know that for balancing P upon Q should be R upon S. So over here P upon Q, P is 100 divided by Q is equal to R is 400 divided by S is 400, right? So basically this is 1, so 100 upon Q is 1, so Q is equal to 100 ohms. So combination of 200 and R should be 100 ohms, right? So 200 and R are in parallel combination, so their effective resistance is 1 upon R equivalent, which is equal to 1 upon 100 is equal to 1 upon 200 plus 1 upon R. So this will be 1 upon 100 minus 1 upon 200, so it is 1 upon 200 is equal to 1 upon R. So R is 200 ohms. R is 200 ohms and that is option 2. Next is question number 126. If X, Y and Z in figure are identical lamps, which of the following changes to the brightness of the lamps occur when switch S is closed? When switch S is open, we have effective circuit as this one. This is lamp X. This is open, so no current will flow through it, and this is lamp Y, something like this. So these are identical, so resistance of both the lamps is same. So effective resistance when switch S is open is 2R. R plus R, 2R, because both are in series. So if this is V, then current through each lamp is V upon R effective, that is 2R. So this is the current when switch S is open. When we are closing switch S, the circuit will be something like this. This is X lamp. Now Z will also be there because now this is a conducting path, so current will flow through this branch also, right? So this will be Z and this lamp is Y. So each having resistor R, but these two R's are in parallel, so their effective resistance will be 1 upon R equivalent is 1 upon R plus 1 upon R. So this is 2 upon R. So R equivalent will be R upon 2. Now this R upon 2 combination is in series with R. So their effective resistance will be total effective resistance. R equivalent is R plus R by 2, that is 3R by 2. Okay? Now resistance is decreasing. So of course current value will increase, right? So total current in the circuit I2 in this particular situation is V upon 3R upon 2, that is 2V upon 3R. So I2 is greater than I1. You can see that I2 is greater than I1. It is 2V upon 3R and it is V upon 2R. So current initially through X was less, now it is increasing. So because of increase in current, the brightness of the X will increase after closing the switch over here. So x increases, option 2 or 3, either option 2 or 3 are correct. Now through y current is V upon 2R, but in the second situation the current is 2V upon 3R up to this point, and after this point current will divide itself equally because these are two equal resistors in parallel. So out of 2V upon 3R, V upon 3R current will flow through the upper lamp and V upon 3R current will flow through the lower lamp. So overall the current through Y is decreasing. Earlier it is 1 upon 2 times V upon R, now it is 1 upon 3 times V upon R. So current through Y is decreasing. So brightness of the Y decreases after closing the switch. So correct answer is option 2. Brightness of X increases whereas of Y decreases. Question number 127. The fundamental frequency of air column is in an open organ pipe is F0. So F0 for open organ pipe is V upon 2L. If it is dipped in water, so that 1/4 of its length is in water. If it is dipped in water, so 1/4 of length is in water, so effective length of the pipe is 3L upon 4 and this end will be closed now. So this will behave like what? One closed organ pipe. Then what will be the fundamental frequency of air column now will be? So this is closed organ pipe, and for closed organ pipe fundamental frequency is V upon 4L dash, where L dash is the length of the closed organ pipe. So it is V upon 4, L dash is 3L upon 4. So we have V upon 3L. But V upon L is 2F0. Using this relation, you can write 2F0 as V upon L. Put it here, bachcho. So you'll get fundamental frequency for this closed organ pipe as 1 upon 3 times V upon L, and V upon L is 2F0. So this is 2F0 divided by 3. So correct answer is option 4. Next is question number 128. Transfer characteristic, that is output voltage versus input voltage for a base bias transistor in common emitter configuration is shown in the figure. For using transistor as a switch, it is used in both regions 1 and 2. If you are using transistor as a switch, you can use it in both regions 1 and 2. So correct answer is option 4. Next is question number 129. A particle of mass m and charge q moves with constant velocity v along the positive x direction. It enters a region containing a uniform magnetic field directed along the negative z direction extending from A to B. So let's say this is x direction and this is negative z direction. So magnetic field is into the plane in this region for us. And this is from x is equals to A to x is equals to B. So width of this region is B minus A. Width of this region is B minus A. Now particle is entering this particular region. We need to find the minimum value of V required so that particle can just enter the region x greater than B. We need to find the minimum value of V so that particle can just enter this region. Okay? So once particle is entering this region, it will experience one magnetic force, and because of that it will perform circular motion, something like this. Let's say it is entering from here. So if it can complete one semicircle like this, it will go back. है ना? If it is just completing semicircle, it will perform a circular motion like this. But if in this region, if it is just completing semicircle, it will go back. Right? So basically the radius of the circle, this is the circular path, it is now like a circular path, but assume that it is a circular path. So radius of the circular path over here, it will go like this. Actually it will not complete the circle. If it is just completing this semicircle over here, so radius of the circular path should be less than greater than equal to B minus A, right? Radius of the circular path should be greater than B minus A, because if it is radius is equal or less than this, the particle will go back like this. We want particle to cross this region, something like this. That means radius should be greater than B minus A, width of the region, right? Now what is radius? It is mv upon qb. So MV upon QB should be greater than B minus A over here. So MV is greater than B minus A into QB. So V greater than B minus A into QB upon M. So this should be the minimum velocity, QB into B minus A upon M, which is option 2. Next is 130, a coil of area 10 centimeter square. This is 10 centimeter square, which is equal to 10 into 10 power minus 4 meter square, has 500 turns. 500 turns, is placed in a uniform magnetic field of induction 50 microtesla, which is equal to 50 into 10 power minus 6 Tesla. It is initially with its plane perpendicular to the field. Coil is something like this, plane perpendicular to the field. It is turned by half rotation about its diameter in 2 seconds. So it is turned by half of the rotation. Matlab complete rotation will be 360 degrees. So half of the rotation, matlab we are turning it to 180 degree. And time taken to turn this one is 2 millisecond. Then the induced EMF in this will be. So induced EMF is minus d phi by dt. So phi initial is BA cos 0 degree. Now area vector will shift there. So angle between B vector and area vector will be 180 degree. So phi final will be BA cos 180 degree. So phi final minus phi initial will be BA cos 180 degrees minus 1 minus BA cos 0 degrees BA into cos 0 degrees 1. So this is minus 2 BA. Induced EMF is minus d phi by dt or minus delta phi by delta t. So induced EMF is minus of phi final minus phi initial divided by time. Right, so this will be minus of minus 2 BA divided by time is 2 milliseconds. Right, so let's put all these values here. So 2 into BA, I need to make some space now where, okay, let's solve it here. 2B, B is 50 into 10 power minus 6 into A is 10 power minus 3 divided by time is 2 milliseconds. So this is 2 into 10 power minus 3, cancel, we have cancelled. So this is, and we have n also, right? So this is into n into 500. So this will be 25 into 10 power 3 into 10 power minus 6. So this will be 25 into 10 power minus 3 volts. So this is 25 millivolt. So correct answer is option 3. Next is question number 131. The value of gamma, that is Cp upon Cv for hydrogen, helium and another ideal diatomic gas X, whose molecules are not rigid but have an additional vibrational mode, are respectively equal to. So gamma is 1 plus 2 upon F. Degree of freedom for hydrogen is 5, for helium it is 3, and for diatomic gas including vibrational modes it is 7. So for hydrogen H2 molecule it will be 1 plus 2 upon 5, that is 7 upon 5. It will be 1 plus 2 upon 3, that is 5 upon 3, and it will be 1 plus 2 upon 7, that is 9 upon 7, right? So gamma will be 7 upon 5, 5 upon 3 and 9 upon 7. So correct answer is option 1. Next is question number 132. On a TP diagram, 2 moles of an ideal gas perform processes AB and CD if the work done by the gas in the process AB is 2 times the work done by the gas in the process CD. So it is 2 times W2. Let's say work done in this one is W1 and work done in this one is W2. Then what is the value of T1 to T2? So work done in isothermal process is nRT ln V2 upon V1, right? So N is same, R is same, V2 upon V1 is same. So ln V2 upon V1 will be same. So basically you can say that W1 upon W2 is equal to T1 upon T2, right? So W1 upon W2 is 2 from this, so it will be 2 is equals to T1 upon T2, so T1 upon T2 is 2, which is option 3. Next is 133. We have two Carnot engines. A and B are operated in series. The first one, A, receives heat at T1 600 Kelvin and rejects it to a reservoir at temperature T2. So this is source for A and this is sink for B. A. The second engine B receives heat rejected by the first engine. So for second engine B, sink of A is behaving like a source of B and rejects to a heat reservoir at temperature T3, which is 400 Kelvin. ठीक है. Calculate the temperature T2 if the work outputs of the two engines are equal. Work outputs are equal, मतलब W1 is equal to W2. Let's say from 600 Kelvin, the heat engine 1 is extracting Q1 heat, doing some work W1 and rejecting Q2 heat. This Q2 heat is extracted by the second engine, doing some work W2, and remaining Q3 heat is rejected at 400 Kelvin reservoir. W1 is equal to W2. Now, if you look at the efficiency is W upon Q, and that is equal to 1 minus T2 upon T1, right? Now, for the first engine, W1 is W because both are same. Q1 is the heat extracted at 600 Kelvin and that is equal to 1 minus T2 is unknown divided by T1 is 600. So this will be 600 minus T2 divided by 600. For second engine, W upon Q2 because input extracted, so extracted heat is Q2 for the second engine and that is equal to 1 minus temperature of the sink, that is 400, divided by T1 is the temperature of the source for this one, that is T2. So this is T2 minus 400 divided by T2. Okay. Now this is equation 1, this is equation 2. Divide these two equations. W cancel, so we have Q2 upon Q1 is equal to 600 minus T2 divided by 600 divided by T2 minus 400 divided by T2. Okay? Now, if you look at the first one, we know that for ideal engine, we have Q1 upon Q2 is equal to T1 upon T2. Q1 upon Q2 is T1 upon T2, so it is 600 divided by T2. But Q2 upon Q1 we need over here, so Q2 upon Q1 will be T2 divided by 600. So it will be T2 divided by 600, right? So let's solve it further. Let's solve it further over here. Let's solve it here. So we have T2 upon 600 is equal to, let's simplify it, T2 into 600 minus T2 divided by 600 into T2 minus 400, right? So T2 cancel, 600 cancel, so we have 600 minus T2 upon T2 minus 400 is equal to 1. So basically 600 minus T2 is equal to T2 minus 400. So 2 T2 is equals to 600 plus 400. So 2 T2 is 1000 Kelvin and T2 is 1000 divided by 2, that is 500 Kelvin. Okay? So temperature T2 is 500 Kelvin, which is option 4. Next is 134. If two rods of length L and 2L are having coefficients of linear expansion alpha and 2 alpha respectively. L alpha, this length is 2L and it is having 2 alpha. These are connected such that the total length becomes 3L. So total length becomes 3L, alpha, 2 alpha, and this is 3L. Let's say effective coefficient over here is alpha effective. The average coefficient of linear expansion of the composite rod will be. So we know that this total length will be 3L into 1 plus alpha effective into delta T, and that is equal to, for delta T increase in length, there will be some increase in length of this rod that is equivalent to increase in length of this rod plus this rod, right? Or you can say that at delta T temperature increase, the total length of this and this is equal to total length of this rod. So length of first rod will be L into 1 plus alpha delta T, plus for second rod it is 2L into 1 plus 2 alpha delta T, right? Let's simplify it. So this is 3L plus 3L alpha effective delta T is equal to L plus L alpha delta T plus 2L plus 2L into 2 alpha delta T. ठीक है, 2L plus L, 3L cancel. So we have 3L alpha effective delta T as we have 2 into 2, 4L alpha delta T plus L alpha delta T, 5L alpha delta T. Delta T cancel, LL cancel. So we have alpha effective as 5 upon 3 alpha. So correct answer is option 3. Next is question number 135. Three lenses L1, L2 and L3 are placed coaxially as shown in the figure. Focal lengths of lenses are given as 30 centimeter, 10 centimeter and 5 centimeter respectively. If a parallel beam of light falling on lens L1 emerging L3 as a convergent beam, okay, so it should emerge L3 as a convergent beam such that it converges at the focus of L3. Distance between L1 and L2 will be, so this is the incoming ray and it should be converging or appear to converge at its focus, converging at its focus, right? So this is focus of first lens, but because of this it will not converge here, it will move something like this parallel, right? Because if you want final beam to converge at the focus of L3, it should be parallel again between L2 and L3 and after that it will converge here, fine. So for the second lens, the beam that is coming towards it should appear to pass through its focus. So basically this distance should be 10 and this complete distance where it is appear to converge is 30. So total distance between this L1 and L2 should be 30 minus 10, that is 20 centimeter, which is 3. So you can solve this types of questions using ray diagram only, that is 20 centimeter. Next is question number 136. Two blocks of masses 1 kilogram and 2 kilogram are connected by a metal wire going over a smooth pulley as shown in the figure. The breaking stress of the metal is 40 upon 3 pi into 10 power 6 Newton per meter square. What should be the minimum radius of the wire used if it is not to break? So over here we have this structure. This is 2 kilogram, this is 1 kilogram, right? So let's find the acceleration and tension first. So let's say this is M1, this is M2, right? So in Atwood machine, we get the value of acceleration as M2 minus M1 into G divided by M1 plus M2, right? So this will be 2 minus 1 into G divided by M1 plus M2, 3. So G upon 3 is the acceleration. Now let's find the tension in the string. So let's pick one block, that is 1 kilogram. So it's 1G will be downward, tension will be upward and acceleration G by 3 will be upward because this system will be moving like this. So let's write second law of motion for this block. So T minus 1G is equal to 1 into G by 3. Net force is equals to Ma. So T minus G is equals to G by 3. So T is 2G by 3. That is 20 by 3 Newton. If you don't want the string to break, then breaking stress of the material should be greater than equal to tension upon area, because on this cross section tension will be there. So tension upon area is equal to stress over here, right? So breaking stress S should be greater than equal to tension upon area. S is 40 upon 3 pi into 10 power 6 should be greater than equal to tension is 20 divided by 3 into area of cross section as pi r square. So 3 pi will cancel this 3 pi. This will be tension upon area, right? 2g upon 3. Okay. I think I made some mistake while calculating something. Let's go back. Okay. So this will be 4g upon 3. Yeah, g plus g upon 3 is 4g upon 3. So this will be 40 upon 3. Right. So this will cancel this. So we have 10 power 6 greater than equal to 1 upon r square. So basically r square should be less than equal to 10 power 1 upon 10 power 6. So r square should be less than equal to 10 power minus 6, or r should be less than equal to 10 power minus 3 meter. 10 power minus 3 meter is millimeter. So r should be less than equal to 1 millimeter. So minimum radius is 1 millimeter, which is option 2. Next is question number 137. If the terminal speed of a sphere of gold, this is sphere of gold, density 19.5 is 0.2 meter per second in a viscous liquid, and density of the liquid is 1.5 kilogram per meter cube. Okay. Find the terminal speed of a sphere of silver, density 10.5 kilogram per meter cube of the same size in the same liquid. So terminal speed is 2r square upon 9 eta into sigma minus rho, where sigma is the density of the body and rho is the density of the liquid. So for the second case, it is V into 2r square divided by 9 eta into sigma is 10.5 minus rho is 1.5, and second case it is 0.2 is equal to 2r square divided by 9 eta into 19.5 minus 1.5 divided. So this will cancel this because r and eta will not change. We are using the same size and same liquid. So this is V upon 0.2 is equal to 10.5 minus 1.5 is 9 divided by 19.5 minus 1.5 is 18. So this is 1 by 2. So V is 0.2 divided by 2, that is 0.1 meter per second. That is option 3, 0.1 meter per second. Next is 138. The work done in increasing the size of the soap film from 10 into 6 centimeter square, that means 60 centimeter square, to 10 into 11 centimeter square, that means 110 centimeter square, is 3 into 10 power minus 4 joules. The surface tension of the film is, this is a soap film, so work done will be 2 times S delta A because we have two surfaces, right? So work done is 3 into 10 power minus 4 into 2, S is unknown, and delta A, change in area, 110 minus 60, that is 50 centimeter square, and we need to convert this centimeter square into meter square now. So this is 50 into 10 power minus 4. So this will cancel this. So we have 50 into 2, 100, right? So we have 3 is equals to S into 100. So S will be 3 upon 100, or it is 3 into 10 power minus 2 Newton per meter, which is option 2. Next is question number 139. A particle executing SHM starts from the mean position. That means its equation can be y is equals to a sine 2 pi by t into small t. At t is equals to 0.5 second, the displacement is half the amplitude. At t is equal to 1 by 2 second, displacement is y by 2, where a is the amplitude, y is equals to a by 2. Its time period is, we need to find the capital T over here. So let's apply this equation for this condition. So y is equals to a by 2, that is equal to a into sine into 2 pi by t into time is 1 by 2. So this will cancel this. So sine something is 1 by 2, है ना? Sine of this will cancel this. Pi upon t is equal to 1 by 2. That means this angle should be pi upon 6 because sine is 1 by 2 at 30 degree or pi upon 6 radian. pi cancel, so we have t is equals to 6 seconds. So time period for this one is 6 seconds, which is option 2. Next is 140. An astronaut is in an orbiting space station above the Earth experiences weightlessness. It is true. It experiences what? Weightlessness. An object moving around the Earth under the influence of Earth's gravitational force is in a state of free fall. Yes, motion under the effect of gravitational force of Earth is known as free fall. So both of the statements are true. A is true and B is true. So correct answer is option 1. Next is 141. 3 identical spherical shells, each of mass m and radius r, are placed as shown in the figure. Consider an axis xx dash which is touching the 2 shells and passing through the diameter of the third. Moment of inertia of the system consisting of these 3 spherical shells about xx dash axis is. So for the upper sphere there is nothing to worry. This is the xx dash axis for the upper sphere. It is diametrically passing through it, right? So for the upper sphere, moment of inertia I1 will be 2 by 3 mr square. For the left sphere, it is tangential. Axis is tangential to the left sphere over here. So we need to apply parallel axis theorem for finding the moment of inertia about xx dash, that is I cm plus mr square, where r is the separation between these 2 parallel axes, right? So it will be 2 by 3 mr square plus mr square, that is equal to 5 by 3 mr square. For the third sphere, again same story. For this sphere, xx dash is again tangential. The distance between these 2 is r, and we need to find ixx dash for the third sphere similarly like this using parallel axis theorem. Right, everything is same. So for the third sphere, you will get ixx dash for the third sphere as same as that of 5 by 3 mr square. So total moment of inertia will be I1 plus I2 plus I3, that is 2 by 3 mr square plus 5 by 3 mr square plus 5 by 3 mr square. So this is 5 plus 5, 10 plus 2, 12. 12 by 3 mr square, and that is equal to 4 mr square. So correct answer is option 3. Next is question number 142. A circular disc of radius r is removed from a bigger uniform circular disc of radius 2r. Okay, we have a bigger disc and its radius is 2r. So I'm considering center as origin. This is x-axis and this is y-axis. So its area is A1 is pi into 2r ka whole square, which is pi 4 pi r square, right? And its initial center of mass will be at the center because this is symmetrical structure. So x1 and y1 both are 0. Okay? Now what we are doing is we are removing certain portion from this disc. So the removed portion has radius r. So for this removed portion, area is A2. Area of removed portion is A2, and that is equal to pi r square. And we need to find that what is the shift in the center of mass of this structure which is left after removing this purple disc. Okay? So for that one, we know that XCM is A1 x1 minus A2 x2 divided by A1 minus A2. A1 is the area before removing, that is 4 pi r square. X1 is the center of mass of the complete disc before removing, which is 0, minus A2 is pi r square, and x2 is the center of mass of the removed disc with respect to the origin. So removed disc is again symmetrical, bachcho. So it will be having its center of mass here, and it is at a distance r from the origin. So it is r divided by A1 minus A2, which is 4 pi r square minus pi r square. So this is equal to minus pi r cube divided by pi 3 pi r square. 3 pi, pi cancel, r square cancel, so we have minus r by 3. So basically new center of mass along x-axis is now here and its distance from the origin is r by 3 along negative x-axis. So the question is, the center of mass of the new disk is AR, that means it is r by 3, that is equal to AR. Then what is the value of A? So you can see that A is 1 upon 3 and correct answer is option 1. Next is 143, the moment of inertia of a wheel of radius 20 centimeter, which is equal to 0.2 meter, is 40 kilogram meter square if a tangential force of 80 Newton is applied on the wheel, its rotational kinetic energy after 4 seconds is. So torque on the wheel is F into R, which is 80 into R is 20 divided by 100, that is equal to 16 Newton into meter. Now we know that tau is equals to I alpha, so alpha is tau divided by I, alpha is tau upon I, so it is 16 upon 40, so that is equal to 0.4. Alright, now after this we can find omega, which is equal to alpha t. Omega naught is 0, so t is time 4 seconds, so it is 0.4 into 4, that is 1.6 radian per second after 4 seconds. So kinetic energy is half I omega square. So it is half I is 40 into omega square is 1.6 into 1.6. So this will be 20, this will be 32 into 1.6 and it will give you 51.2 joules, which is option 2. Next is 144, a sphere of mass m moving with a constant velocity hits another stationary sphere of the same mass. If A is the coefficient of restitution, then the ratio of speed of the first sphere to the speed of the second sphere after collision will be. So let's say after collision this sphere is moving with V1 and this sphere is moving with V2. So we need to find V1 upon V2. So let's conserve momentum first. So initial momentum is mu is equal to final momentum is mv1 plus mv2. M cancel. So u is equals to v1 plus v2. Now what is coefficient of restitution? Velocity of separation, which is v2 minus v1 divided by velocity of approach, that is u. So u from this equation is v2 minus v1 upon e. Put it here. So you will get v2 minus v1 upon e is equal to v1 plus v2, or v2 minus v1 is ev1 plus ev2. So you can write v2 minus ev2 is equal to v1 plus ev1. Okay. So let's solve it here. You can take v2 common, so this is 1 minus e and that is equal to v1 into 1 plus e. So v1 upon v2 will be 1 minus e divided by 1 plus e, that is equal to option 1. Now next is 145. A system consists of 3 masses m1, m2, m3 connected by a string passing over a pulley P. The mass m1 hangs freely and m2 and m3 are on the rough horizontal table. The coefficient of friction is mu. The pulley is frictionless and of negligible mass. The downward acceleration of mass m1 is m1, m2, m3 are given as m. Right? So if you look at this one, this mg force will try to pull this system in the downward direction, but the friction on block m2 and m3 will try to pull back this system in the opposite direction. So what is the friction on this one? Mu mg. What is the friction on this one? Mu mg. So mg is responsible for pulling this system in this direction, whereas mu mg and mu mg, these two forces, are responsible for pulling this friction, blow this system in the opposite direction. So net force in this direction will be mg minus mu mg minus mu mg, and this is the net pulling force. Okay? So this is mg minus 2 mu mg, and net pulling force divided by mass will give you acceleration. So total mass of the system is m plus m plus m, that is 3m. So m cancel. So we have 1 minus 2 mu into g divided by 3 as the acceleration of the m1. 1 minus 2 mu into g divided by 3. That is option 3. Next is 146. A boat crosses a river of width 200 meter in the shortest time and is found to experience a drift of 100 meter. So for shortest time you should cross it directly perpendicular like this, and because of the velocity of river you will be having some drift. So this will be the final position of u, and this drift is given as 100 meter, and this is velocity of swimmer with respect to river. The time taken in this case is t. If the same boat is to cross the river by shortest path, the time taken to cross will be. So over here, if you look at this one, the horizontal velocity is responsible for the drift in t time. So Vr is 100 upon t, whereas for crossing this river we need vertical velocity, and we have only one vertical velocity, that is Vsr. So Vsr, velocity of swimmer with respect to river, is 200 divided by t. Now in the second situation, we have the same boat or swimmer and we want to cross the river by the shortest path. So if river is flowing like this, we should start something like this at some angle theta with Vsr speed so that we can reach here, like this. So let's take the components. So this is Vsr cos theta and this component is Vsr sin theta. So net velocity of the swimmer with respect to ground will be the vector summation of these two. So we have Vsr cos theta in the upward direction, Vsr sin theta in the left and Vr in the right. So for zero drift, if we want to cross it with the shortest path, drift should be 0. We should reach directly opposite. For zero drift, Vsr sin theta should be equal to Vr. What is Vsr? 200 by t. 200 by t into sin theta should be Vr. Vr is 100 by t. 100 cancels, tt cancels. So we have 2 sin theta is equal to 1. So sin theta is 1 by 2. So sin theta is 1 by 2, that means angle is 30 degree. That means this is Vsr cos 30 degree, which is root 3 by 2, right? Now this velocity is responsible for crossing 200 meters. So time taken in the second case, t dash is 200 divided by this velocity, which is Vsr root 3 by 2. But Vsr is 200 by 2 t. So this will be 200 divided by 200 by t into root 3 by 2. So this cancels, so we have 2t upon root 3. So time taken to cross in the second case is 2t upon root 3, which is option 4. Next is question number 147. A rocket of 6,000 kilogram mass is set for vertical firing if the exhaust speed of the gas is 1000 meter per second. We have mass as 6,000 kilogram. The rate of mass of gas ejected to supply a thrust, we need to find dm by dt, needed to give the rocket an initial upward acceleration of 20.2 meter per second square is. So let's solve it, guys. Let's solve it. I think we can directly apply m into g plus a is equals to Vr into dm by dt. M is 6,000, g is 9.8 plus a is 20.2 is equal to Vr is 1000 into dm by dt for rocket propulsion, right? So this is 21 plus 9, 20.2 plus 9.8 is 30. So this will be 6000 into 30 divided by 1000 is equal to dm by dt. So this will be 180 kilogram per second, which is option 2. Direct formula based. Next is question number 148. We have two particles move at right angle to each other. Their de Broglie wavelengths are lambda 1 and lambda 2 respectively. This is particle P1, this is particle P2, lambda 1, lambda 2 wavelength. The particles suffer perfectly inelastic collision. The de Broglie wavelength lambda of the final particle is. So over here we know that lambda is h upon p, so p is equals to h upon lambda. So momentum of the first particle is p1, which is h upon lambda 1. Momentum of the second particle is p2, that is h upon lambda 2. Now both of this momentum are perpendicular to each other, so net vector summation of these two momentums will be under root of p1 square plus p2 square, which is equal to under root of h upon lambda 1 ka square plus h upon lambda 2 ka square. Now this is the total momentum of this system, so final momentum should also be same. So final momentum of the particle is p and its wavelength is lambda, then this should be equal to final momentum, and that is equal to h upon lambda. So let's square on both the sides, so we'll get h square divided by lambda 1 square plus h square divided by lambda 2 square is equals to h square divided by lambda square. H square cancel. So we have 1 upon lambda square is equals to 1 upon lambda 1 square plus 1 upon lambda 2 square. Next is 149. A time dependent force F is 3t square plus 6t acts on a particle of mass 18 kilogram. If particle starts from rest, the work done by the force during the first 3 seconds will be. So we can apply work energy theorem over here. So work over here is equal to K final minus K initial. K initial will be 0 because particle is starting from rest. Initial kinetic energy will be 0, half mv square. So work done is equals to K final, but it is P square upon 2m. Where P is the P final square upon 2m, where P is the momentum of the particle, right? So, we know that dP upon dt is equals to F. So, dP will be F dt, integrate on both the sides. So, we'll get P final minus P initial is equal to integration F dt, that is 3t square plus 6t dt. Right, and the limits are from 0 to 3 seconds. So this will be 3t cube divided by 3 plus 6t square divided by 2 and 0 to 3. 3 cancel, so this is t cube, that is 3 cube plus this is 3 into 3 ka square, so this is 27 plus 27, 54. Now put it here, so it is 54 into 54 divided by 2 into m, m is 18. This will give you 27 into 3, that is equal to 81 joules. Next is consider the following statements for a particle in one-dimensional motion and identify the correct statement. With 0 speed at an instant may have non-zero acceleration at that instant. ठीक है, 0 speed may have non-zero acceleration. Yes, motion under gravity में highest point पे particle का velocity 0 लेकिन acceleration नीचे की तरफ, right? At highest point velocity of the particle is 0 but acceleration is vertically downward, that is g. So statement 1 is true. Statement 2, the instantaneous speed is always equal to the magnitude of instantaneous velocity. Yes, at a particular instant the magnitude of instantaneous velocity is equal to instantaneous speed. So this is also true. So both A and B are true. Both of the statements A and B are true. I hope it is clear.
Hi everyone, welcome to Infinity Learn. My name is Palak and we will be discussing mock paper of chemistry. Let's begin. Which of the following compounds are polar? So we are given these compounds A, B, C, D and E and F. 6 compounds are given to us. CCl4 tetrahedral dipole moment will be 0. For a polar compound, we need the dipole moment will be non-zero. BCl3 trigonal planar, it will also be 0. Coming to ammonia, ammonia is this. So ammonia is pyramidal, here dipole moment is not equal to 0. So first answer is C. So let's see the options in which I have the option C. So I can eliminate this. A and B are non-polar. So the options in which I have A and B will also be omitted. So this will be omitted, this will be omitted. My answer will be option number 2. Now let's check for the D1, SO2. SO2 is also bent. We have one lone pair here. So this is also a polar compound. Here the dipole moment is non-zero. Coming to the other cases, benzene is non-polar, SO3 is also non-polar. So from the options directly we can eliminate the options and get the correct answer with the help of ammonia only. And ammonia is a very common compound which is polar. Next question. The correct statement about the element, we are given a particular electronic configuration and we need to identify which of the following is correct. Remember, in every exam, please read carefully. Sometimes they are asking correct, sometimes they are asking incorrect. So please be sure about that. Correct statement we need to find. It belongs to S block. So let's see this. If I go to the atomic number, what will be 6 plus 18, 18 plus 7, it will be 25. So we know that first the electron enters in the 4s and then electron enters in the 3d. So this will not be the correct option. Next, it has the highest oxidation state of +7 in its organic compound. So we can say that it will be having highest oxidation state. Yes, it will give electron from 3d as well as 4s because the energy level of 3d and 4s is almost same, so it will be similar. So it will give all these 7 electrons, and we know that it is already manganese, and manganese shows the oxidation state of +7. So this is the correct option. Now let us see the other options as well. It is a non-metal? No, it is a D block element. D block elements are metals generally, and manganese is a known metal. So this is again wrong. It belongs to third period of the modern periodic table. No, it belongs to fourth period of the periodic table. So this is wrong. What is the correct answer? Correct answer is option number 2. So again a very easy question. The ratio of velocities of methane and O2 for the molecules having de Broglie wavelengths. Now what is the formula for de Broglie wavelength? This is the basic formula for de Broglie wavelength, H upon mv. H is the Planck's constant, m is what? M is the mass, v is the velocity. Now I have same de Broglie wavelength for two molecules, so I can equate lambda 1 equal to lambda 2, right? So I can write H upon m1 v1 is equal to H upon m2 v2. Now I have H here, it will be cancelled. So in the other step I will write m1 v1 is equal to v2 m2. I need to find the ratio of what? Ratio of velocity. So I need to find the ratio of v1 by v2. So it will be what? m2 by m1. What is this m? Mass, molar mass I would say. So what is this? m2 is for oxygen. So it will be 32 by 16. Methane it is 16, so 32 by 16, cancelling we get the value as. So what is the correct answer? The correct answer is option number first. Also people are generally confused. Many of you have marked the answer as 1:2 because they are generally getting confused कि m1 by m2 आएगा या m2 by m1 आएगा. Next question. A container of volume V contains an ideal gas with pressure this atm. Now if the container is divided into n equal compartments, what will happen? See, in case of this we know that the equation is PV is equal to nRT, where n is the number of moles of gas, P is the pressure, V is the volume. Now in this case I am dividing the my container into an equal apartment. So I can say that I have a one big box of volume V, I am dividing it in smaller parts. So will it it affect the actual volume? No, the actual volume of the will remain same, the container will remain same. So what will the pressure? The pressure will be same only, the pressure will be P, it will not change. So what is the correct answer? Correct answer is option number 1. Next, at 0 degrees Celsius, ice and water are in equilibrium. So we have ice and we have water, right? Solid form of water is ice. We are given delta H, the values of delta G and delta S we need to calculate. Now remember, at equilibrium the value of delta G is 0. So the value of delta G will be 0. So in every option it is given as 0. Now let us talk about delta S. So at equilibrium there is a formula to calculate delta S. What is this? Delta S is equal to delta H by T. What is this T? T is the transition temperature. So what is the transition temperature here? 0 degrees Celsius. So will I be dividing it by 0? No. Then it will be infinity, no, not defined. Remember, we need to convert this degree Celsius into Kelvin. So don't forget that. So I'll just substitute the values. 6 kilojoule per mole divided by temperature 273.15 Kelvin. Right. So we can see that the value is in joule per mole per Kelvin. So I'll convert. So it will be approximately 6000 divided by 273.15. From here you can calculate, and what will be the correct answer? You can calculate, the value will be, it will be positive, it will not be negative. So what will be the correct answer? You can mark 155. So correct answer approximately you can calculate, and the answer will be 21.9. Right? Correct answer is option number 3rd. Next, for a homogeneous mixed reactions, we are given particular reaction. What all data is given? Delta H, delta S. At what temperature the reaction is at equilibrium? So in this case, what you need to write? You need to actually substitute the values in this expression. We know that to calculate or to identify whether our reaction is spontaneous or non-spontaneous, we are using the expression of gives free energy. Now, here at equilibrium what happens? At equilibrium the value of delta G is equal to 0. So my expression will change. Delta H is equal to T delta S. Now I need to calculate the temperature. So temperature will be what? Delta H by delta S. So what all the values are given in the question? Always keep this thing in mind. Generally in every question the value of delta H is in kilojoules. Conversion karna hai, don't forget to convert. So it will be minus 30. So this is in joule, this is in kilojoule. So in order to get this value in pure Kelvin, so I need to do what? I need to convert the values. So it will be 1000 joule per mole. While solving, please write the unit, take care of the unit, so that there are less chances of the question getting wrong. So what will I do? I'll just cancel these. Joule and joule will be cancelled. I will be getting the values in Kelvin. Now there was no tough calculation. It was what? Minus and minus will be cancelled. It will be 300 Kelvin. Now remember 300 degree Celsius फटाफट जल्दी से बहुत लोगों ने कर भी दिया होगा mark. Why? Because we got the value as 300. They will immediately mark the answer as 300. No, don't do this. Don't repeat this mistake. It is in Kelvin and these options are in degree Celsius. So what you need to do? You need to convert Kelvin to degree Celsius, minus 273. So answer will be option number 4th, 27 degree Celsius. Clear? Next question. the mass of carbon consumed giving only CO2 in the production of 270 kg of aluminium metal from bauxite by Hall-Héroult process. So this process we are actually studying in metallurgy. So this is a question from metallurgy combining it together with mole concept. So we are actually preparing what? We are preparing aluminium metal from bauxite using what? Carbon. So what will be the reaction? Carbon will react with Al2O3 and it will give me what? It will give me pure aluminium plus CO2 gas. Now we need to balance this reaction. So we after balancing you can see it will be 4 aluminium and what will be this amount of CO2? It will be 3 CO2. You can check, is this balanced? 3 carbon, 3 carbon, 6 oxygen, 6 oxygen, 4 aluminium. Done. So now this is the reaction of what? Hall-Héroult process. Now we need to identify how much carbon will be required for the preparation of 270 kg of aluminium. So you can see, you have to apply mole concept here. We can see that 3 mole of carbon is giving what? 4 mole of aluminium, right? From the reaction we can see that 3 moles of carbon are giving me 4 moles of aluminium, because I need to actually find the relation. I need to find the mass of carbon and the mass of aluminium is given to me. Now what will I do? So it is in terms of moles. So can I convert in terms of mass, because this value is given in mass only. So 3 into what? 12 gram of carbon will react to give 4 into 27 gram of aluminium. Is it? This will be only, I am converting moles to mass, so I am multiplying by molar mass. 3 into 12 will give me this data. So I can write here 36 gram of carbon is giving me what? 27 into 4 will be 108 gram of aluminium, right? 36 gram of carbon will give me 108 gram of aluminium. Now what I need to find? I need to do the reverse. So I can write that 108 gram of aluminium is formed from the reaction of 36 gram of carbon. So I need to find 270 kg will be formed from how many grams of or kg of carbon. So simple unitary method, how can I find? I will be 36 by 108 into 270 kg. So these are in grams, these are in kg, so final value will be in kg. So it is 3. So dividing it, you will be getting the value as 90 kg. So what is the correct answer? The correct answer is option number 3rd, 90 kg. So here we have combined actually 2 concepts, metallurgy and mole concept. I hope this is clear. Coming to the next question. Identify incorrect statement from the following. So we need to identify the incorrect statement here. Zinc can liberate hydrogen gas when it reacts with NaOH. Yes, it reacts with NaOH to form zincate, sodium zincate plus hydrogen gas. Next, iron chromate is the catalyst involved in water gas shift reaction. So this is also true. When we are doing water gas shift reaction, the catalyst is iron chromate. Now this is also important. In the yesterday's mock test, sir might have discussed this, that H2D2 have same type of bond length. Next, this is very common answer and which is a very common statement. Hydrogenation of oil. In hydrogenation, we are never using manganese. We are using nickel or palladium or platinum. Manganese is never used in hydrogenation. So this is the incorrect statement. So the correct answer is option number 4. At 298 Kelvin, the extent of ionization of this amount of ammonium hydroxide to ammonium ion is this one. So what is this extent of ionization? We are given the value of alpha, that is 0.1%, or we can say 0.1 by 100. This is the value of alpha. The extent of ionization is alpha. They are talking about ammonium hydroxide to ammonium ion. Now if I have ammonium hydroxide, it will give me what? It will give me NH4 plus OH minus ions, right? Now if it is a weak base, so that is why the alpha is given to us. This is alpha. So in such cases we need to find pH. Now to calculate the pH, what I need the concentration of, it will be minus log of, sorry ma'am, what are you doing? OH minus nahi, it is H minus, because I am writing pH. Shit, what I did. I can't undo this, no problem. So I'll write it again. What is given? Alpha is given to me 0.1% or 0.1 by 100. So NH4 ions are weak, this is a weak base, so it will give me NH4 plus OH minus ions. Now this is weak, it will give me what? The OH ions. So initial concentration C 0 0, after some time this will be C minus C alpha and C alpha. I need to calculate the pH. What is pH? pH is equal to minus log of H plus ions, right? From here I am getting OH minus ions. So what is the concentration of OH minus ions? It will be C alpha. What is the value of C? 0.1 into 0.1 divided by 100. So this will give me the what? This will give me the concentration of OH minus ions. So I'll write it as approximately 10 raised to power minus 4. Now from here I'll be calculating pOH. It will be what? Minus log of, so it will be 10 raised to power minus 4 ka when I'll be taking log, it will be 4. So this is what pOH. We know that at 298 Kelvin, pH plus pOH is what? 14. So if this is 4, what will be pH? pH will be 10. So what is the correct answer? Correct answer is option number 1. Clear? Next, equilibrium constant. We are given 3 reactions. We need to identify the equilibrium constant for this reaction. 4A gives me D. Now let's see. You can see that in the product, in the desired reaction, A is double. So I'll just do what first step I'll do. I'll multiply this reaction by 2. So if I am multiplying this reaction by 2, my K will become what? If I am multiplying it by a certain number n, that n goes in power. So my reaction will become 4A gives 2B, and what will be the K? How will it change? It will become K1 square. Right? Next. Now I got 4A. Now B is there, B is also here, but in my final equation I don't have B. So I need to eliminate B. So to eliminate B, what can I do? I can subtract. But what to subtract? So first I'll do one thing. I'll reverse this reaction. So it will become 2B and gives me C. And how will my K change? It will become 1 by K2. Right? Now if I add these two reactions, so what will happen? On adding these reactions, I will be getting 4A will give me C. Right? And what will be the new K? It will be K1 square by K2. And it will be K1 square by K2. I hope this is clear to everybody. So this is the second step I have done. Now last, 4A gives D here. Here it is giving C. So I need to cancel this also by using this equation. So you can see if I divide this equation by 2, I will be getting C 2C gives 2D. So it will be, I'll be changing this. So I'm dividing this by 2. I'm dividing this by 2. So what will be my new reaction? C gives D. Right? And what will be the K? K will be K3 raised to power half. Because I can say I can multiplying it by half. So the half will go to power, so it will be this. Now this is my reaction and this is my reaction. If I take this 1, if I make this 2, am I adding 1 and 2? Then what will happen? If I am adding 1 and 2, then what will happen? The C will be cancelled and I will be getting my desired reaction. This will be my desired reaction. Now what will happen to the K? Nuclear equilibrium will be, when I am adding reactions, what happens to the K? K will be multiplied. So it will be K1 square into K3 raised to power half by K2. So this will be my desired equilibrium constant for this reaction. Let's see if this is matching. K1 square, K3 half, K2. Yes, option number 2. So what is my correct answer? The correct answer is option number 2. So are you all with me? Understand? Is it clear? So what is the correct answer? Correct answer is option number 2. Next, coming to the next question. Identify the correct trend regarding alkali metals. So lithium is the most abundant element. This is not true. Sodium is the most abundant element. Okay, so this is wrong. Density, when we are moving down, the density increases. So this is the correct option. Now coming to the conductance, lithium is heavily hydrated. So it will not have the highest conductance, it will be having the least conductance. So what will be the order of conductance? It will be reverse order. Conductance will be higher for cesium, followed by rubidium, then potassium, then sodium. Again melting point, reverse order. Melting point will be highest for lithium, then followed by sodium, etc. So what is the correct trend? Correct trend is here for the density. So what is the correct option? Option 2. Next question. Okay, ammonia plus O2 in the presence of platinum is giving me A. So what happens? This is a direct reaction when we are combining nitrogen with oxygen. First I am getting NO. When NO is again reacting with oxygen, I am getting NO2. This will give me NO2, right? When NO2 is reacting with water, so I will be getting two compounds, HNO3 and again NO. So what is the correct answer for this question? The correct answer for this question is option number 1. Clear? Next. Okay, when copper reacts with hot concentrated solution of H2SO4. So copper is reacting with H2SO4. Now we know that this is a very strong oxidizing agent. The oxidation state of sulfur here is +6 and here it is 0. Now what will happen? Copper will change to copper 2+. This is an oxidizing agent. It will increase the oxidation state of copper to +2 and it will decrease its oxidation state. So it will decrease its oxidation state to form SO2. Now here two possibilities are there for the products SO2 and SO3. But in SO3 the oxidation state is +6. So here it will not be behaving as an oxidizing agent. So what is the correct option? Correct option is option number 3rd, SO2. Clear? Deacon's process for the preparation of chlorine. So Deacon's process is a well-known name reaction. This is the answer when HCl is reacting with. So it is a process of formation of chlorine from HCl plus CO2 in the presence of COCl2 to give me what? Cl2 and H2O. So this is the reaction of Deacon's process. The hybridization of central metal in these are respectively. So you might have studied this in classes as well. Nickel CO4. Here what is the oxidation state of nickel? It is 0. So we have D8 electrons. 3D8 electrons. 1, 2, 3, 4, 5, 6, 7 and 8. Although this is a strong ligand, okay, D8 then 4s2. D8, 3D8, 4s2. This is for nickel in 0 oxidation state. So it is a strong ligand. So what will happen? It will do the pairing and these electrons will be paired. So after pairing, what will happen? Here we have 10 electrons. Now S will be empty and P will be empty. So this is 3D, 4S and 4P. So what will be the hybridization? CO, 4 CO ligands are here. So we need to accommodate 8 electrons. 8 electrons will be accommodated here. They will come here. And what will be the hybridization? It will be sp3. So for this NiCO4, the hybridization will be sp3. So this eliminates one option. But ma'am, this is not worthy because all options are, rest of the options are still there. They are not eliminated. Now, let's check. This is also a strong ligand and this is a weak ligand. Now, what will happen? In the case of strong ligand, pairing will take place. Now, here we have a strong ligand, Cn. So, here nickel is in +2 form. So, what will happen? Nickel is in +2 form. So, in nickel +2, what will be the number of electrons? 3d8, 4s, 0. So, I have 8 electrons here and it is a strong ligand, so pairing will take place. So, 8 electrons will be like this. Now, this D will be empty. D will be empty and S. So, what will happen? We need to accommodate 8 electrons. So, 1, 2. What will be the hybridization? It will be DsP2. So, here the hybridization will be DsP2. So, now this is also eliminated. Hybridization will be DsP2. Here what is the other possibility? It is a weak ligand. It will not undergo pairing. Pairing will not take place. Now, pairing will not take place. So, here there will be electron and the hybridization will be what? sp3. Because pairing will not take place. So, what is the correct answer? Correct answer is option number 2. The hybridization of this cannot be DsP2 because for DsP2 we need to have one empty or vacant d orbital which is not here. So, what is the correct option? The correct option is option number 2. PPH3RHCl is a catalyst. What is this catalyst? This is Wilkinson catalyst. Wilkinson catalyst. Now, it is a very common catalyst. Where it is used? It is used in the hydrogenation. Now, remember, hydrogenation it is used. So, dehydration will be wrong. This will be wrong. Hydrogenation of oil. No, for hydrogenation of oil, we are using nickel. Here, it is used in the hydrogenation of alkene. So, what is the correct answer? Correct answer is option number 2. Next, pick up the true statement about the complex compound with the formula. So, we have this complex compound which has the formula. So, we can see that this compound actually is in the form of metal A3 and B3, right? So, this will definitely form what? Facial and mer isomer. Remember, this will form facial and mer isomer. So, what are these? This is what? This is one isomer and what will be the other isomer? So, this is facial isomer and this will be mer isomer, right? So, you can see the complex can exhibit facial or mer isomer. This is the correct option. Let's see the other options as well. The IUPAC name, Triamine cobalt, no. The ligand is also chlorine, so it has to be here. Triamine, trichlorido or chlorocobalt, right? So this is wrong. Complex can show optical isomerism. No, it cannot show optical isomerism. The hybrid state of cobalt is DsP2. No, we have a octahedral complex. For octahedral complex, we can't have the hybridization. It has to be either D2sp3, or sp3d2. Why? Because here we have the complex is octahedral and DsP2 is in square planar where the coordination number is 4. So it is the wrong option. So what is the correct answer? Correct answer is option number 2. Next question. In aqueous solution, the ion which is colored. So a very basic definition of the colorful complexes, you might have covered this in D and F block elements. Now if we have D1 to D9 electronic configuration, D1 to D9 electronic configuration, our compounds will be colorful, colored. If I have D0, D10 electronic configuration, they will be colorless. Now coming to here complexes, Fe2 plus, it will be 3D6,4S2. This is what? Fe normal. Fe2 plus will be this. So it will be 3D6,4S0. It comes in this category, so it has to be colored. Now let us talk about the other complexes. It will be what? 3D10, 4S0. So it comes under this category. Zinc 2 plus, again 3D10, 4S0. Scandium 3 plus, it will be 3D0. Right? 3D0, 4S0. So all these will come in the category of colorless and Fe2 plus will be colorful complex. Blue baby syndrome is caused when? So this is a simple theoretical question. Blue baby syndrome is caused due to the formation of or excess of nitrate ion. So you know this will be wrong. Now when we have the concentration of nitrate ion more than what? More than 50 ppm, then we can say that it will be blue baby syndrome. So here we have less than 50. So what is my answer? Answer will be option number 2. Clear? So the correct option is 2. I hope all of you are getting your answers correct. And you all are solving. That is most important. You know, also the other important thing is please learn from your mistakes. After giving the test, you might be realizing that you know this question is very easy, but we could not solve in the exam because of the exam pressure. So release all the pressure from your mind and feel free to solve the question. Now, which of the following mixture shows negative deviation from the Raoult's law? So whenever our solution is non-ideal solution, we have two categories, positive and negative deviation. In negative deviation, what happens? The AB interactions are stronger than individual AA and BB interactions, right? Now, in negative deviation means that after forming a solution, they are forming the bonds which are more stronger than the individual AA and BB bonds. So, coming to this first example, CHCl3 and so this is actually a very common example. Here what happens? This has what? Dipole-dipole interactions. Because it is a polar compound, here it also has dipole-dipole interaction. Now when we are forming a solution, they are actually doing what? They are actually forming hydrogen bonds. So I have this chloroform, what are they doing? They are forming hydrogen bonds. So as a result of which, you know, you can see as a result of which this hydrogen bond, this AB interactions will be stronger and they will show negative deviation from the Raoult's law. Here if I talk about this compound, here I have this non-polar compound, right? So here I have dispersion forces, here I have hydrogen bonding. So the solution will never have the hydrogen bonding. So it will show what? It will show positive deviation from the Raoult's law. right? Similarly non-polar, so again जो solution बनेगा after forming the solution, then also they will be non-polar, so they will form ideal solution. Similarly here, similar ideal solution. So what is the answer? My answer is option number 1. What is my answer? My answer is option number 1. Next, metal has an FCC lattice, okay. The edge length of the unit cell is this, the density is this, the molar mass you need to calculate. So very easy question, direct formula based. What is the formula of density? Z into M, A cube into Na, Avogadro number. So substitute all the values, I need to calculate the value of M. So it will be what? D into A cube Na by Z. What is the density given to us? So density is 2.72 gram per centimeter cube. A cube, picometer. Now this is in picometer, so 404. Now this is in centimeter. So we need to convert this picometer into centimeter. You can convert, it will be 10 raised to power minus 10 centimeter, and we'll be doing cube into Na. What will be Na? Avogadro number 6.02 into 10 raised to power 23 per mole, divided by what? Divided by 4. For FCC we know that the value of Z is 4 for FCC. So what is the correct answer? You can calculate, you will be getting the correct answer. Okay, after calculation, so what will be the molar mass approximately? I think it will be approximately 27. You can also check. Next, the limiting conductivities for NaCl, KBr and KCl are given to us. We need to find the limiting molar conductivity of NaBr. So in this case, please remember, if I need to find for NaBr, then the terms which are having Na and Br will be added, the remaining terms will be subtracted. Now, so I have Na here and Br here. So I'll add these and I'll subtract this. So let me write the expression. So limiting molar conductivity for NaBr is equal to, it will be NaCl plus KBr minus KCl. So what will be the value? NaCl 126 plus 152 minus 150. So it will be what? 152 minus 152, it will be 128. So what will be the correct answer? The value will be 128. So what is the option? Option 1 is the correct option. Time taken for 90% completion for a first order reaction. So in my classes also I used to tell student, first order reaction is very important. So you should, remember all the formulas related to the first order reaction, because generally we are getting questions on first order only. Time taken for 90% completion of the first order reaction is 2 minutes. Now the time required in seconds for 99.9% of the same reaction to be completed is. So there is a relation, direct relation between T half and T 99.9. So T 99.9 is equal to thrice of T 90%, right? So you can see it will be 2 into 3, 6 minutes. 6 minutes means 6 into 60, that is 360 seconds. So what will be the answer? The answer will be 173. Answer will be option number first. Clear? This is the direct relation between T 99.9% and T 90%. If not, suppose you didn't remember the formula, then what will you do? You will just calculate by using this formula. We have this formula for first order reaction, which you always remember, A upon A minus X. So you can write T99.9 and you can calculate for T90. From here, K will be same because we have this particular reaction. For a particular reaction, from there you can calculate and you can find the relation. So what is the correct answer? The correct answer is option number 1. Next question. Critical temperature of SO2, CH4 and H2 are this. Okay? The increasing order of the extent of physisorption. So actually the extent of physisorption is directly proportional to the critical temperature. Extent of physisorption is directly proportional to critical temperature. Okay. So whenever temperature is more, it will be more. So temperature is more in SO2 case. So it will be more for SO2, then followed by methane, followed by hydrogen. So what will be the answer? Answer will be option number 2. Clear? What will be the answer? Answer will be option number 2. That is 174. Next question. So such type of questions we can get in from biomolecules chapter. So sucrose is made up of glucose and fructose. So see, glucose, fructose, this. So for first definitely second will match, P will match with second. So you can see the options, directly you will get the answer. But in this paper this happened, generally it never happens. Generally we are able to eliminate 1 or 2 options only. Let's check for the other. Starch. Starch is made up of 2 amylose and amylopectin. So it is a polysaccharide which is made up of amylose and amylopectin. Next, maltose. We can see maltose Q is matching third. So let's check. Q is made up of what? Both are alpha D glucose. Lactose is made up of beta D glucose and beta D galactose. So you need to remember them. You are getting them directly. But here this question was easy because the option choices were really easy. So sucrose, everybody knows, inversion of sucrose gives me what? Glucose and fructose. So everybody remembers this. So I hope every one of you have given the answer correctly. Which of the following is wrong with respect to proteins? So during denaturation of protein, what happens? Only secondary and tertiary structures are getting disrupted. Primary structure will remain as such. So it destroys 1, 2 and 3 degrees. So this will be the wrong statement. Our answer has to be this. Now let us check the other options as well. Change in pH. Yes, because of the change in pH, denaturation of proteins take place. Heating a protein destroyed 2 degree and 3 degree. This is also correct. Denaturation can be reversible. This is also correct. So always remember, during denaturation what happens? We are destroying only 2 degree and 3 degree. That is secondary and tertiary structures. Primary structures will remain same. What is the correct option? First, pick the correct option for the polymerization of phenol and formaldehyde. So phenol and formaldehyde forms a polymer by condensation polymerization. This is correct. Linear polymer is known as Novoloc. So you can see these direct reactions are given in NCERT. You know that linear polymer is known as Novoloc. Slightly blanching take place. Branched or cross-linked polymer is known as Bakelite. Now Bakelite is used for non-making. No, this is wrong. Non-stick cookwares. Non-stick cookwares we are using Teflon. Bakelite is used in making switches, right? This is Teflon is used in making non-stick cookwares. Clear? So what is the correct answer? Correct answer is option number 4. Next question. A very easy question. Carbocation order of decreasing stability. So everyone knows what is the order of stability? 3 degree, 2 degree, 1 degree. Carbocation is electron deficient. If we have more and more electron donating groups attached to the carbocation, then it will be more stable. So this is the order. So what will be the order? This is 3 degree, 2 degree and 1 degree. What will be the answer? Answer will be C, B, A. So what is the answer? Answer is option number 1. Next question. Which of the following is not a component of photochemical smog? So the answer is chlorofluorocarbon. Chlorofluorocarbon is a pollutant which causes ozone depletion. It is not responsible for for photochemical smoke. All these are ozone, methane, and formaldehyde, HCHO, PAN, this is PAN, acrolein, ozone, all these are responsible for, causing photochemical smog. But CFC is not responsible for this smog. Chlorobenzene on treatment with sodium in dry ether gives biphenyl. The name of the reaction is a very common reaction. When we are combining our aryl benzene with sodium in dry ether, the reaction is Fittig reaction. Double, we are getting biphenyl. So when I am reacting chlorobenzene, we take 2 molecules of chlorobenzene with 2 sodium in the presence of dry ethers. This is the Wurtz reaction, but Wurtz reaction, what we take? I take this alkyl, not aryl. So what will be the product? Product will be diphenyl or biphenyl. In Wurtz reaction, it is the speciality of Wurtz reaction that the number of carbons are getting doubled. So this is biphenyl, so the reaction is Fittig. What is Wurtz Fittig? In Wurtz Fittig, I am taking one as alkyl halide, other as aryl halide. So it will be Wurtz. Gutterman, it is a reaction from diazonium salt. So this is not Gutterman reaction. Wurtz reaction same. Alkyl halide reacting with sodium in dry ether. We are getting the alkane with double number of carbons. Very easy question. The best preferable reagent for the conversion of alcohol to pure alkyl halide. What is the answer? SOCl2. All these are chlorinating reagents. They will replace OH by Cl. But why are we using SOCl2 or thionyl chloride? Because here the side products are gases, so our main product can be separated easily, or we can separate the main product, can be isolated easily. So what is the correct option? Correct option is third. I hope you all are with me, you all are able to solve these questions. These are very easy questions. Keep your josh high. Next. Meta-dihydroxybenzene is known as, so this is meta-dihydroxybenzene. This is what? Resorcinol. Okay. This is resorcinol. What is quinol? Paracrisol is ortho, and this is catechol is different. No, catechol is ortho and crisol is different. So our answer is resorcinol. Which of the following carbonyl compound are most reactive towards nucleophilic addition reaction? So for nucleophilic addition reaction, carbonyl compound pe where it takes place on the carbonyl carbon, because of our carbonyl carbon is electrophilic, right? So this is the center at which this is partial positive, this is partial negative. So here this is the electrophilic center. Now if I have any electron donating groups here, then what will happen? There will be steric hindrance, and as a result of which the reaction will be slow. So which of the following will react faster? This, because here steric hindrance is very less, there is no steric hindrance, and you can see that the carbonyl carbon is electrophilic here, as you can see, right? So my answer will be methanol. If I have the CH3 group here electron donating, so it will decrease the electrophilicity of the carbonyl carbon, and nucleophile will come with less ease. So what will be the answer? The answer will be very easy, methanol. The acidic nature is highest in. Okay, so this is ortho. We have phenol here, we have phenol here. We know that if we have a electron withdrawing group attached to our group, then we can say the acidic character will be more. But here we have the COOH here, so this will be more acidic than phenol. We know that carboxylic acid, acidic character of carboxylic acids is more than the acidic character of phenol. The carboxylic acids are resonance, the carboxylate ion is resonance stabilized. Now generally people think, ma'am, we have more resonating structures for phenoxide ion, but still carboxylic acid have more acidic character than phenol, because here the negative charge is stabilized on the 2 oxygen atoms. But in case of phenol, the negative charge is, is resonance stabilized only on 1 oxygen atom. Here we have 2 oxygen atom in case of carboxylate ion. So that is why even though it has only 2 structures, but it is more stabilized by resonance in comparison to the phenoxide ion. So what is my correct answer? Ma'am, the compound which contains COOH. So answer is option number 2. Carbylamine test. Oh my god. Carbylamine test is given by 1 degree amines. 1 degree amines, it is given by 1 degree amines. Now, it is not given by 2 degree or 3 degree amines. The amines can be aliphatic as well as aromatic. Okay, both of these can give carbylamine test to form what? To form isonitrile or isocyanide. Okay, now 2 degree will not give this test and 3 degree will also not give this test. So out of these we need to identify which is primary, which is not. So this is primary, this will give, this is also primary, this will give. Now let us talk about this. Generally tertiary, people are thinking that it has to be 3 degree, but this is not. Let me draw the structure, tertiary butyl amine. So here this is amine. No, you people generally students have confusion. So this is what tert-butylamine. So here it is also primary. So this will give the test. On the other hand, I have this diethylamine. So CH3, CH2. So this is diethylamine. It is 2 degree. So it will not give the carbylamine test. So just this information will be helpful to you. This also I have discussed in the PYQ series of amines, this question, similar question, okay. So I hope this is easy, very easy question. Correct structure of I3 minus ion. So we can interpret the structure using VSEPR theory, right. I3 minus ion, I can write this as I2 minus. Now it will be easy, this will be my central atom and this will be my surrounding atom. So first what will I do? I need to calculate the number of bond pairs and lone pairs. So number of bond pairs are 2. Bond pairs will be 2 because surrounding atoms are 2. Now I will calculate the value of lone pair also. Lone pair. Lone pair, how can I calculate the number of lone pairs? Number of electrons on central atom, that is 7. Negative charge, so I'll add 1 electron, minus number of electrons in bonding. So 1 iodine is forming 1 bond, 2 will 2 form. So this divided by 2. Divided by 2. So what will be the answer? 8 minus 2 by 6 by 2, it will be 3. So here I have 3 lone pairs and 2 bond pairs. So please check 3 lone pairs and 2 bond pairs. So this will not be the value, this will not be the. Now you can see this is what? TBP, trigonal bipyramidal. In TBP what happens, if we have 3 lone pair, they will always go to the equatorial position and the axial position will be occupied by the iodine atom. So what is the correct answer? The correct answer will be option number first. Clear? Next, the excited electron of hydrogen atoms emit photon of wavelength lambda and returns to the ground state. The principal quantum number of the excited state is given by. So we know that whenever an electron is getting excited, it goes to the higher level, but it will not remain for a longer time to the higher level and return back to the lower level, right. So what will be the wavelength? The wavelength will be given by this formula. Right? What is N1 here? N1 is the number of, N1 is the initial level. And N2 is the what? Final level. So it is going to excited state and coming back to ground state. So we can say that the value of N1 is 1. What is N2? N2 is the excited state. We don't know which is the excited state. So as you can see from the options, we actually need to calculate the value of N2 in terms of all the values. So I'll write R 1 by 1 minus 1 by N2 square. So what will I do? I need to calculate the value of this. So I'll just rearrange this. It will be 1 by lambda R minus R by. Next, what can I do in the next step? Or I could have done one more thing. I can keep this here, alpha R. So it will be more easy. So it will be lambda R minus 1 is equal to 1 by N2 square. Okay? I can do this. Now, I can also rearrange 1 minus lambda R by lambda R is equal to 1 by N2 ka whole square. I'll just reverse this order. So I can say N2 square will be equal to lambda R upon 1 minus lambda R. What will be the value of N2? It will be square root of this is a moderate question. Can you see which option is matching? So it will be 1 minus, yeah. Is there any option which is matching? I think, let's check. 1 minus N2 square. So this will be, oh, I have done one mistake here. It has to be what? R minus, this will be 1 minus 1. So it will not be like this. It will be, that is why it's not matching. It will be like this. 1, just a second. What we did, forget this step. I kept this here. So it will come here. It will be what? 1 minus R lambda. It will be like this, right? So in the next step, what I'll be getting, this will interchange lambda R minus 1. So I hope you understood what is the problem, right? Here it will change, so it will be reverse. So value will be, it will be minus 1, again lambda R minus 1. So what will be the final value? Understood? So this is the answer. Answer is option number 4. The only mistake I made was I need to take this, ये यहाँ लेके आना चाहिए था, मैंने उल्टा कर दिया था. So I hope it is clear now. So what is the option? Answer is option number 4. The root mean square velocity of a diatomic gas is V. So what is the value of root mean square velocity V RMS? It is under root 3 RT by M. Now, for a particular gas, it is V. When the temperature is doubled, the molecules dissociate into atoms. So we have two conditions. Now initially we have temperature T and we have a mass of a particular molecule. Now what happens in second condition, when temperature is changing to 2T, what happens? The molecule will dissociate into atoms, right? The molecule will dissociate into atoms. So what will happen? What will happen to the molecular weight? Molecular weight will become half. So I can write in other condition, I can see that V1 upon V2 is equal to, it is what? Under root T1 upon M1 into, it will be what? V2 will be here, so T2 by M2. Right? Is this the case? We need to calculate the value of new RMS. Now V1 is what? V for us. So V upon V2 will be equal to, this will be T, this will be M. Now what is happening? Temperature is getting doubled, and what about mass? Mass is getting half. So it will be again this. So let's cancel this. So V1 upon V2 will be approximate under root 1 by 4. So it will be 1 by 2. So what happens to the V? What is the value of V2? V2 will be 2V. Very easy question. So what is the answer? Answer is option number 3rd. Clear? When the molecule is dividing into atoms, what will happen? The molar mass will become half. That's the change I have done, and this is the final answer. 189. Hydrogen combines with chlorine to form HCl. So hydrogen is combining with chlorine, right, to form HCl. This is the ratio. So okay, let's consider the other element. Nitrogen is also combining with hydrogen. Sorry, nitrogen, not sodium, is combining with hydrogen to give me NaH. If sodium and chlorine combines with each other, what will be the ratio of their masses? Now, if Na is combining with Cl, what will be the ratio of the masses? So, you remember law of constant proportion. According to law of constant proportion, the elements are always present in the ratio of their atomic masses, right? So, what is the ratio here? It will be 1:35.5. Here it will be 23:1. Now, when they are combining in the ratio, so what will be the ratio of atomic masses? 23:35.5. Simple. What will be the answer? The answer will be option number 1. Cl:2Na hota, but this is not the case because cation is always coming first. So, what will be the answer? 23:35.5. All the mixtures can act as buffers except. Okay, so remember, in case of buffer, we must have a weak acid or a weak base. Clear? Here I have this weak acid. Okay. This is also weak, this is also weak. Weak as what? It is a salt, right? This is not a weak acid. Now, this is a weak base. KOH and HCl, both are strong. Strong base, strong acid. Can strong acid and strong base form buffer? No. They can never form buffer. So, what is the correct answer? Correct answer is option number 4. Okay. Now, see, we can also see that when they will react, KOH will react with HCl to give me what? KCl plus water. Right? Let's calculate the moles. 200 ml into 0.025. Here 100 into 0.15. Which value will be more? This will be approximately 5 into 10 raised to power minus 3. And this will be 15 into 10 raised to power minus 3 moles. So, what will be in excess? This will be in excess acid. HCl will be in excess, right? So, this will be a somewhat an acidic medium. This will not act as a buffer. So, what will be the answer? Answer will be option number 4. Next, coming to the second group, metal hydroxides, means they are talking about calcium hydroxide or beryllium hydroxide and the trend. What happens to the solubility? Here we have this OH minus ions. So, what happens to the solubility, thermal stability and basic strength? So, all these are going to increase or decrease. What will happen? Do you have any idea? What will happen to the So, generally what happens, the solubility, thermal stability and basic strength all will increase down the group from beryllium to the last element, barium. So, you can see the first, second, third all will increase down the group. Which of the following compound does not form during the hydrolysis of HCF6? So remember one thing, during hydrolysis what happens? Oxidation state remains same. This is a very important funda. You know, it will not change. So during hydrolysis, oxidation state of xenon will not change. So what is the oxidation state here? It is +6 here. So let us calculate the oxidation state of this compound. So now what is the oxidation state here? +6. Why? How? X minus 6 will be equal to 0. Here x minus 4 minus 2 will be equal to 0. So here it will also be +6. Because you know already how to calculate the oxidation state. X minus 2 minus 4 will be 0. So in all the cases we have the oxidation state +6. So these cannot be the answers. So answer has to be this. Let's check. X minus 2 minus 3 is equal to 0. What is the oxidation state? +5. So this will be our answer, because what happens on the hydrolysis of XeF6, we will be getting the compounds which have same oxidation state of xenon. So here the oxidation state is changing, so this has to be the answer. Answer is option number 4. Now, for a cell reaction involving 2 electron transfer, the standard EMF of the cell is, what is this? This is the standard EMF of the cell. So E0 is given to us as 0.295 volt. 2 electron transfer means they are telling us that the number of electrons which are transferred are 2, so N is equal to 2. What is the value of equilibrium constant? Now how can I calculate the equilibrium constant? What is the formula? E0 cell is equal to 0.059 by N log of K. This is the value, right? So what is the value here? This is 0.295, 0.059 by 2 log K equilibrium, right? You will solve this. So into when log K equilibrium will be equal to 0.295 into 2. Okay, so solving this will be approximately 10 times of this, it will be this. Now, so the value of 10 raised to power log K equilibrium is 10. So we can just equate, no? We know that it will be equal to log of 10 raised to power minus 10. Then what will be the value of K equilibrium? What is the answer, ma'am? What is the answer? Answer is, it will be plus 10 or minus 10? Just a second. What is this? This is positive or negative? Right? This has to be positive because here the value is positive. So, ma'am, it will be plus 10. So, what is the answer? Answer will be option number 2. I hope this is clear now. Don't do these mistakes. When we are taking G, no, G in G expression we have negative sign. But in the expression of K equilibrium with E cell we don't have any negative sign. So, remember this. Don't make these mistakes. Now 194. Total number of products formed by the monochlorination of butane. Butane is this. So monochlorinated products actually depends on the different types of hydrogens we have. Here I have this one hydrogen and the other kind is this. We don't have any other type of hydrogens here because if I do the chlorination here it will be identical and same. So what are the two types? What are the two types of product which I can get on monochlorination? So Cl2 H nu, it will be ClCH3 and the other will be CH2 CH2 CH2. So what are the two types of products I will be getting? I will be getting only two type of products on monochlorination of N-butane. Because I have only two types of hydrogen, this one and this one. Okay, phenol on NaOH, then what will happen? it will give A. So very simple reaction, phenol is acidic. On reaction with NaOH, it will form ONA. H will be replaced by sodium. Now on reaction with CO2 and H plus, it will form what? CO2 and H plus. This is a common name reaction. Now first question pura pad lo. Ma'am, question mein we need to identify the incorrect statement and we are given this options. So ultimately we had to do the reaction. So this will give me what? This will give me benzoic acid. This is a name reaction which is what? Kolb's reaction. This is a Kolb's reaction. You know, when we are converting phenol to benzoic acid. First we will be reacting with NaOH, so ONA, it will react with CO2 to give me this. Now, what is this? What is happening here? One more thing. Also, this is Kolb's reaction. Here, we are getting orthohydroxybenzoic acid or salicylic acid. Now, don't forget this. Next, we are doing what? CH3CO altosis and anhydride. So, it will, this will be COOH. And what will change here? यहाँ पर this is acidic anhydride, CH3COO, and what will happen? This will change, and what will be the product? The product will be here OCOCH3. This will be my final product. So let's see, the preparation of B from phenol is Kolb's reaction. So this is A and this is B. This is a Kolb's reaction, it is correct. B is stream volatile. B is stream volatile. Yes, this is also correct. C has a free OH group of B. What is C? This is C. C has a free OH group? No. It do not have a free OH group. C can be used as an anti-inflammation, analgesic, etc. So this is a very common analgesic antipyretic. Okay, what is this? What is the name? You can guess the name. Okay. So what is the correct answer? Correct answer is option number 3rd. Next, CH3CHO reacting with dilute NaOH, it will give me X. Find the number of sp3 hybrid orbitals of the carbon atoms in the organic compound. Now generally they are asking what is the product, but no, they are not asking what is the product. What will be the reaction here? Please answer, what will be the reaction here? Come on, yes. So many of you have given the answers which I think will be correct only. Very common reaction, aldol condensation. We have alpha hydrogen here, it is reacting with dilute NaOH. It will give me aldol product. So I'll just write the product. This will be the product, right? Aldol condensation after, heating. So number of sp3, so this is sp2, sp2, sp2. Now what will be the number of sp3 hybrid orbitals here? So we have this sp3 hybridized carbon. In sp3 hybridized carbon, how many are sp3 hybridized orbitals? There will be 4, right? sp3 hybridization, we are getting 4 sp3 hybrid orbitals. So what will be the answer? Answer will be option number 1. Next, okay. When this is diazonium salt, it is reacting with HbF4. What are we getting? X. So X will be this N2BF4, the salt, right? Now, when we are doing heating, when we are heating this directly, we are getting this compound, fluorobenzene. But if we are not heating it directly, we have this compounder X, we are reacting it with NaNO2 followed by heat. Then what will happen? This is a direct reaction from NCERT. Okay. Now we are getting Y and Z. So After reacting with NaNO2, it will give me NO2 plus N2 gas plus this product, right? Now what is Y and Z here? Here Y and Z can vary, it can be anything, but here nitrogen is not given to us. So what is Z for us? Z is 2. So the option is option number 2. Next question. Mole fraction of toluene in the vapor which is in equilibrium with a solution containing benzene and toluene. So in the solution we have two components, benzene and toluene. This can be 1, this has to be 2. Having 2 moles each, when the vapor pressure of pure benzene and toluene are 120 and 80. So what we need to calculate? We need to calculate the mole fraction of benzene in the vapor phase, toluene in the vapor phase. So we need to calculate the value of Y2. Now what is Y2? It will be P2 divided by P. Now ma'am, what are these two? I have taken 1 for benzene, 2 for toluene. I need to calculate the mole fraction in vapor phase, right? So it will be mole, this is what? Partial pressure of toluene divided by total pressure. So this will be P2 naught into mole fraction divided by P. Now we need to calculate everything. First we will calculate the number of moles. So number of moles are directly given. So N1 is 2, N2 is also 2, right? So I will be calculating the mole fraction. It will be N2 upon N1 plus N2. It will be what? 2 by 4, 0.5. P2 naught is what? The vapor pressure of the pure substances. For pure toluene it is 18. So I will just write here. It will be what? P2 80 into 0.5 divided by. We need also to calculate the total pressure. What will be total pressure? Total pressure will be P1 plus P2. Right? So I will just calculate 120 into 0.5 plus 80 into 0.5. This will be 60 plus 40, 100. So it will be 100 torr. I substitute the values here, 100 torr. 40 by 100 will be 0.4. So is the 0.4 given in the options? Yes. What is the answer? Answer is option number 3. Next, which of the metal is not present in German silver? So German silver is an alloy of copper, zinc and nickel, right? So this is Sn is not present here. So what is the correct answer? Correct answer is Sn. Standard enthalpy of formation is 0 for all the following. So standard enthalpy of formation is 0 for all the elements who are in standard state, right? So the standard state of phosphorus is what? White phosphorus, right? When phosphorus is in white phosphorus form, it is the standard state. So red phosphorus will be correct answer. Graphite, rhombic sulfur, oxygen, all these are present in the standard state, right? So it will be answer. Answer is option number 1. So that's all from my side. I hope you understood all the solutions. All the best for your test. Thank you.